change in the schedule of today. This is because of a condolence meeting organized at NII jointly for 3 issues. That is at 11 o'clock. So we will have Professor Ranabir talk right now, and then we will have a tea break, and then we will start at 12:15 with lecture by Mandar Deshmukh, and then Jithender Reddy, and evening we have Dinesh talking on the practical aspects, NMR spectrometer. So you have a time, means we will, after his lecture, we will have a break, and then at 12:15 we will start with Dr. Deshmukh. Okay.
Good morning. Hello. Good morning, everyone. Welcome to 4th day of the workshop. I would like to introduce our first speaker, Dr. Ranabir Das. Dr. Das has completed his integrated PhD in physics from IISc Bangalore. He was a postdoc fellow at National Cancer Institute, Maryland, USA. Currently, Dr. Das is working as associate professor at National Center for Biological Science, Bangalore. Today, Dr. Das will talk about application of NMR in enzyme kinetic study. So, without any further ado, I would like to welcome Dr. Das.
Hello. Hello, good morning. Can you hear me? Okay, thanks. Very good morning to you. I hope you are having a wonderful session in this workshop the last couple of days. Today, I would be talking about looking at enzymes. If any of you are interested in studying proteins, enzymes, finding out inhibitors of enzymes, because as you know, several enzymes and kinases are responsible for various dysregulations and malfunctions in human cells that lead to cancers and stuff like that. So, we will look at monitoring the enzyme kinetics by NMR, and there are certain advantages of using NMR for this process, which we will discuss, and then go through a very specific example that was perfectly performed in our lab, and probably that will give you an idea of how it can be employed in a very effective manner. So the basic question is these enzyme reactions, which probably all of you have studied in your class 12 or early school, is the chemical enzymatic reaction where there is a substrate and an enzyme, and the substrate binds to the active site of the enzyme, and then there is a catalytic effect going on, and the enzyme is releases the product after converting the substrate into the product. And these enzymes carry out a variety of functions inside the human cell, for which a lot of our cellular processes are responsible. So the design of inhibition kind of goes in this way, is that if you really want to inhibit the function of this enzyme, then you design an inhibitor that binds to the active site and does not allow the substrate to interact at the active site. And these, although I am talking about these inhibitors which are binding at the active site, there are also allosteric inhibitors that bind to somewhere else in the enzyme, but still through conformational changes perturb the binding of the substrate to the active site, and that way also act as inhibitors. These probably you know as competitive inhibitors versus non-competitive inhibitors. The way that we measure enzymatic reactions typically is we measure the Michaelis-Menten curve, which also you probably have looked at in the textbooks in school or colleges, where we measure the reaction rate of a enzymatic reaction, the velocity, against the substrate concentration. And if we plot it beyond a certain substrate concentration, this plot is going to saturate. The 50% rate at which the velocity is half of the maximum velocity gives the Km, which kind of indicates the affinity of the substrate at the enzyme active site. The two cases that I discussed about this competitive versus non-competitive inhibitors are the competitive inhibitors bind at the active site and they reduce the Km, but they do not affect the Vmax. And the non-competitive inhibitors do not bind at the active site, they bind out somewhere else, but they still inhibit the reaction and they affect the Vmax without affecting the gain. So the question is, how do we measure these velocities and plot the Michaelis-Menten curve? The way that typically we can do is we can carry out a chemical reaction, biochemical reaction, for example this, which is here is the enzyme ubiquitin complex. I will talk about it in a few slides, but I wanted to with the introduction. For example, so you can say that this whole thing is an enzyme, and there is another molecule of ubiquitin which is a protein that acts as a substrate. It binds to the active site and forms this product, and then the enzyme is released. So this is a typical reaction, enzymatic reaction, which you can carry out in tubes, Eppendorf tubes, and then take out time points at each aliquots and run a gel, and then measure the rate of the ubiquitin or diubiquitin formed here, which is the product. So with time you can see the product forming, and you can see this free form of the E2 starting to appear from this complex, which was the active enzyme complex. So we can do this, and then we can measure the velocities of this at different concentrations. So these are, with time I am plotting the amount of product formed in y-axis, and x-axis is time at various concentrations, and I am seeing that the slope of this is increasing, which is essentially the velocity is increasing with time, and beyond a certain point it is saturating. So with this we can plot this kind of a reaction rate versus substrate concentration, and then we can look at the Vmax and the Km values of this particular reaction, and then when we design inhibitors, what changes happen at this kind of a Michaelis-Menten curve. The problem with this is that the reaction is happening and you are taking multiple time points, aliquots, and this gives rise to certain amount of handling errors. For example, pipetting, how much volume are you picking up, at what exact time are you picking up. Instead of this, if we run this reaction in a real time manner, in the sense that you start the reaction and you don't perturb the system at all, reaction is happening and you are able to monitor the rate of the product formation in real time without doing any pipetting or handling the system at all. If we could do that, then there are certain advantages, because it has been shown by various groups, research groups previously, that it is much elegant for the person who is doing the experiment. Also, it's much less error prone. So the errors contribute to measuring the velocities, it's much lesser. The question is, can we use NMR? Because there are some other techniques, biophysical techniques also, which you can use. For example, fluorescence spectroscopy. Can we use NMR to do this kind of experiments? One of the advantages of using NMR is a tag-free method. So we are not putting any sort of additional compounds or tags onto the protein or the enzyme of interest, so that we can do not perturb the system at all. So this is an advantage which is not present in, for example, fluorescence spectroscopy, which will need some sort of a fluorescent tag to be introduced into the system. So if we could do this by NMR, then you would do these experiments with much less error. The question is, how do we do that? What is the principles that we should follow? The principles are very simple, that we can run this reaction in the NMR tubes and continuously monitor by NMR the rate of the product formation. The important thing that we should keep in mind is that the resonance frequencies of the reactants, thus the substrate, and the product should be different. Otherwise, you will not be able to distinguish between the reactants and the products. Either their resonance frequencies are different, they have entirely different chemical shapes, or if there are chances of overlapping, and we use isotope labeling and isotope editing experiments to do these experiments such that we can observe only one of the species. For example, you are interested in the rate of the product formation, so you can look at the rate of the product formation. With time, the reactants or the substrate is going to decrease and the intensity of the product is going to increase. So we'll start the reaction, carefully measure the intensity of the product resonance, and in NMR the line intensity is is directly proportional to the amount of the product. So there's nothing in between. So if we monitor the intensity of the product, then we can correlate to the amount of the product formed, and then we can measure the velocities. One thing that we have to be careful is that in NMR intensities are relative, so nothing is absolute. So one should have some sort of an internal control, something that is always present in the sample, which does not overlap with these resonances, and you can use that intensity to calibrate all the other intensities as these intensities are changing with time. And then we plot this against the time, the intensity of the product formation, and we get the velocities. To apply this, I will take one example from our lab. Our lab is heavily invested in studying post-translation modifications. Post-translation modifications are modifications that happen to a protein after the protein is translated from the RNA to the protein. These post-translation modifications, we know that protein structures are very important for their function, but post-translation modifications is an additional component, an additional dimension by which these functions and the structure of the protein can be regulated. So it is very important to understand how protein post-translation modifications happen and how do they regulate their function. There are a variety of post-translation modifications that one can study in protein. The range of post-translation modifications in proteins and how they affect the cellular functions is widening and increasing and becoming very complex as well as interesting to study. One of the ones that we are studying very intensely in the lab is this modification, which is ubiquitination. In some of the modifications, you would see that there are small chemical groups being added, like a methyl group, a glycosyl group, or a acetyl group. In the case of ubiquitination, it is actually an entire protein that gets added to the side chain of a protein, and that regulates the function or the fate of this protein. The way that it happens is a multi-step enzymatic reaction. First, ubiquitin gets added to an enzyme called E1 with the help of a ADP-dependent reaction. Then ubiquitin is like a bucket transferred between different people. It gets transferred to a second class of enzymes called E2, and then gets transferred from E2 to the substrate with the help of a different class of protein ligases called E3 ligases. This reaction continues and a chain of polymeric ubiquitins are formed on the substrate. One of the major functions of these ubiquitin chains is to degrade the substrate. We need to maintain a certain amount of proteins inside the cell. We cannot allow proteins to go beyond certain levels inside the cell. So the regulation of this protein levels inside the cell, one of the major ways to do it is using ubiquitination, which then targets these protein substrates for degradation. I'll talk a little bit later about these classes of enzymes called de-ubiquitinating enzymes, which basically cleave ubiquitin chains from the protein substrates and then relieve them from this degradation mechanism. This is another way of sort of controlling the system such that unwanted proteins which were tagged for degradation can be rescued. The way that ubiquitin chains form is through an isopeptide bond between the lysine side chain and the C-terminal end of ubiquitin. So this is the substrate, for example, and it has a lysine side chain which is exposed, and it forms an isopeptide bond with the C-terminal tail of ubiquitin. Now ubiquitin itself has 7 more lysines inside it, and so these chains can now be extended using those lysines and the C-terminal tail of another ubiquitin to form these type of polymeric chains. Depending on the lysine that is used for the ubiquitin to extend these chains, these different types of lysines can be seen. M1 is a extension that happens to the N-terminal methionine side chain. So these chains, depending on their type of lysines that they are used to extend this chain, have a different structural conformation, and because of their different structural conformation, they bind to different adapter proteins inside the cell. and consequently these different chains have different functions inside the cell. For example, a chain that is built up using K63 is used for DNA damage and innate immunity related purposes, whereas the K48 types of chains is specifically related to protein degradation mechanisms. So if we look at this, this ubiquitination process is particularly complex because there are multiple enzymes, but to make things simpler for today's discussion, we can particularly look at this reaction, which is the E2, which has a ubiquitin on it, and the protein substance. Look at these polymeric chains that are forming. So this is what I am showing here. The E2 with the ubiquitin conjugated to it gets transferred, the ubiquitin gets transferred to the protein substrate with the help of a protein class of enzymes called E3. Now the question is, in this elongation process, you would see that the chain is formed by the one ubiquitin whose lysine now attacks the active site and then consequently forms the takes the second ubiquitin, and this is how the chain is elongated. Now if we ask the question that how are these chains, how is the specificity of the chains been built? How does the enzyme decide what kind of chains will be built onto the substrate and what determines that? If you carefully think about this process, you would understand that this must be depending on some critical interactions around this lysine, which is attacking the active site, and this active site of this E2 enzyme, which is favorable for this lysine only to attack the active site and not the other lysines that are present on this ubiquitin. These favorable interactions will allow this lysine to attack the active site and extend this chain using only this lysine, which could be K48, K11, K63, whatever. So the question is, how do we understand what are the favorable interactions? The way that in the field that we do it is either we use, so these reactions are very transient and very difficult to really stabilize and look at their structures. One way that we can do is we do some chemical mimetics such that we can stabilize this intermediate and solve its structure to understand what are the interactions. Or the other way to do that is we can model this E2 and the ubiquitin complex, intermediate complex, and see what are the favorable interactions for it. But in either case, whether you do it by solving a structure or you do it by modeling, you have to finally look at this intermediate, the contacts, the favorable contacts between the E2 and the ubiquitin, validate them by enzymatic assays, and in which step again NMR can be very helpful. So we have to validate these inferences from either one or two by kinetic assays, and the question is, can we use NMR to do that? So for example, the model of the enzyme substrate complex that we modeled using some chemical NMR softwares looked like this. So this is the enzyme that we are looking at. Its name is UBE2K. It forms only K48 type diubiquitin chains. The question is, why is it only forming K48 type ubiquitin chains where there are 7 more lysines present in the ubiquitin? In the complex, we see that these are the orientations of this ubiquitin, which is already attached to the enzyme, and this is the orientation of the ubiquitin that is attacking the active site and then taking the this ubiquitin away and forming the diubiquitin. If we look at this complex, we see several critical interactions present between the ubiquitin lysine that is coming to the active site. For example, with this aspartic acid D124 at the active site. Remember, the active site is this C92, but there is an aspartic acid which also forms a hydrogen bond with this K48, and there are other interactions in and around this K48 which are also favoring this close conformation for the substrate to attack the enzyme. So how do we validate that these things are at all important or not? So what we did was we used NMR to measure ubiquitination rates in real time. I am talking about ubiquitination rates here, but you could measure any enzymatic rates using NMR by similar principles. Remember, we said that we need to somehow make sure that the resonances of the product can be monitored such that it is not convoluted with any other resonances in the spectrum. Now, this is particularly difficult for the ubiquitination reaction because the incoming substrate is of ubiquitin. The product is a diubiquitin, which also has a very similar structure to ubiquitin. So, we have to find out some way to distinguish it, and it's just not looking at the spectra, we cannot distinguish between the product and the reactant. So, we have to use NMR isotopes and some sort of clever NMR editing techniques to specifically look at the product. The one that we used was, we noticed that the lysines of ubiquitin, side chain lysines, these amide bonds, exchange rapidly with solvent and are invisible in the physiological conditions. So, the side chain lysine amine very difficult to observe in NMR in non-physiological conditions, which is pH around 7.2. If we go at very low pHs, for example pH 2 and at extremely low temperature, Junji had shown that it is possible to see these lysine side chains, and they appear in this ppm around 33 to 34 and 7.8 in the proton dimension. Yeah. Well, in the normal pH, I mean this normal temperature and this thing that you take the spectra, these are invisible, you cannot see them. They are rapidly exchanging with the solvent. So, they are not even visible, so there is no question of folding. But if you go to this temperature, for example 2 degrees and pH 5, then the exchange drops and then you can see them. Yeah. Yeah, it will fold, it will fold. So what Junji did was, he used a very selective pulse to just only excite between 33, 30 and 35. So he is not exciting the backbone amides at all. Yeah, selective pulse. So remember, this pH is very low, it's not the physiological pH. Temperature is extremely low, around 2 degrees centigrade. So we cannot really run ubiquitination reactions in these conditions, because these are not physiological. So we have to find some other way to look at these things. But one of the interesting things is, when this amine now forms a diubiquitin and becomes an amide, then it becomes a normal amide bond, which we typically see in normal backbone amide spectroscopy experiments like HSQC. So now it becomes a typical amide, like we have amides at the backbone of proteins, and now we can look at them by NMR easily. So, for example, which was appearing at 33 ppm, now it comes around 128 ppm. So now it is possible to actually look at this diubiquitin, which is the product, exclusively in the spectrum. For example, and then monitor the intensity of this growing peak with time, and then just plot that to get the velocities. For example, if we, this is the first method that I will discuss, which is the simplest method but has some difficulties with time resolution, is that we have a acceptor ubiquitin. Remember, this acceptor ubiquitin is going to attack the active site, and this is the other molecule that is attached to the enzyme. And with the formation of the product, this NH2 will form an amide bond, and you can observe this in the NMR spectra by a normal N15 proton HSQC that we carry out to characterize any proteins at the initial step. And this is the peak that comes, and over time, at 0 minute, this peak is not present because we have not started the reaction. After you start the reaction, around 6:48, you will see this peak coming up, and it is actually, we are just taking a snapshot, but over time, if you keep just monitoring this intensity of this growing peak, you can see a peak coming up, which is directly related to the amount of product formed and can be used to measure the velocities. So this is the first method, that is, you just label your ubiquitin with N15 labeling, standard isotope N15 labeling, and carry out a conventional 2D HSQC and measure the intensity and get your product rate of formation, plot the velocities, get the Km and the Vmax of the reaction. The problem is there could be overlap between other HSQCs, other amide resonances in the HSQC onto this peak. Fortunately for ubiquitin, this did not happen, but for other proteins, for example, this could be a problem. When you are looking at the ubiquitin formed on a substrate, for example, and we are looking at the backbone amide HSQCs, you would see that the resonances of the substrate may overlap with this, and it could be a problem. The other problem is that the temporal resolution is low. You still have to take an HSQC, which for our case was giving an temporal resolution about 30 minutes or so. So if you are monitoring a reaction with only 30 minutes resolution, you may not be happy with that. You might want to get more points to with a smaller time resolution to monitor the reaction rates more effectively. So there is another method that you can use that, which uses a little bit more of clever isotope labeling, is that we label this accepted ubiquitin with N15, and that's okay that if all the other amide bonds are also present in the HSQC spectrum, and we label the donor ubiquitin with carbon 13. Now after this amide bond is formed, remember this molecule is entirely only N15 labeled, and the donor ubiquitin is entirely labeled with only C13. So now after this bond is formed, there is only one group of 3 atoms which are bonded by this kind of a bond, which is the proton, nitrogen and carbon, and the nitrogen and the carbon are labeled. So now if we just do a simple C13 and 15 edited 1D, then this is the only group of atoms that is going to show a resonance in the spectrum, because none of the other molecules have this C13 and 15 at the same place. Either is going to be either N15 here or C13 here. And this is the experiment is actually basically simplified form of the 3D HNCO experiment that we regularly carry out to do backbone assignment experiments. And if we now monitor this 1D version of this, then we again see a rising peak formed in this spectra. And this, remember, this is just a simple one-dimensional NMR experiment, and the time resolution of this is around 10 minutes now, because remember we are not doing a two-dimensional experiment now, which is taking multiple transients. This is a one-dimensional experiment, and so that simplifies the time resolution, brings it down to 10 minutes only. And that you can use to again monitor the reaction rates. There is a third method that is also uses a sort of a different type of isotope labeling. This labeling is called the labeling of N15 epsilon lysine. So you can get isotopes which will only label the lysine side chains and none of the backbones. And if you do that and don't label this at all, at the end of this form there is going to be amide formed, that's true. But remember, these lysine side chains are invisible at normal physiological temperatures and pHs. So they exchange rapidly and they are not visible. But when this forms an amide bond, then they start becoming visible, because now they are a simple amide molecule atoms. So if we do a simple N15 edited proton detected 1D, then we see this, this is the only resonance that comes up in the spectra, and we can again monitor these intensities to monitor the rate of the reaction form. The resolution of this experiment in our hands were around 3 minutes or so. So now it is possible to monitor the rate of the ubiquitination reaction formed in under 3 minutes. And for example, we use this to show that we could look at the formation of K63 type diubiquitin in the presence of this activator called E3, which increases the rate of the reaction, or in the absence of the activator E3. And the time resolution for this experiment was 5 minutes, but in certain conditions we can go down to 3 minutes or so. So what I presented today are 3 methods. You can use either or not, depending on what are the labeling that you are comfortable with. In one case, we used uniform N15 labeling and collected HSQCs. The time resolution was lower here. The other one we used a N15 labeled acceptor ubiquitin and a C13 labeled donor ubiquitin, and then we carried out a simple 1D form of HNCO, and the time resolution was lower here. And or we could label the N15 side chains of the molecule ubiquitin only, and then we collect a simple 1D again, and then we can monitor the rate of the reaction formed. So coming back to this complex of the E2, the donor ubiquitin and the lysine 48. Yeah, that's another way to look at it. So if you form this, if you label this molecule C13 and then look at the C13 molecule when it becomes an amide. Do the CN experiment, basically C13 detection and F15 on the indirect dimension or something, or edit it. Yeah, direct C13 set. So now we wanted to find out that are these contacts at all important or not, these hydrogen bonds that we saw. So what we did was we mutated this aspartic acid to a arginine, and we in fact see a very dead drop in the rate of the reaction. So this is the Michaelis-Menten and this is the rate of the product form, and you can see that the rate of the reaction decreased drastically. When we substituted this arginine to a opposite chain, which is a glutamic acid, the rate of the reaction was reduced because it has some interactions, indirect interactions through aspartic acid, but not as much. And this cysteine, this serine forms a hydrogen bond with Q49, and when we substituted this serine 85, again the reaction completely dropped, which makes us understand this interaction between Q49 with S85 is very critical for this region of the substrate to come to the active site, and this is what stabilizes this reaction and gives rise to the catalytic activity of the enzyme. And this is what makes this specific, because this Q49 is not present before the lysine, other lysines in ubiquitin, which makes this K48 very specific for this enzyme, and that leads to the extension of the polymeric chain in using a very specific lysine, which then leads to protein degradation. Now coming back to this deubiquitinating enzymes, which cleaves this ubiquitin chains from the substrate, we can also monitor the activity of these deubiquitinating enzymes on this polymeric chains using the same set of reactions or the techniques that we saw. So the trick that we used was we made this diubiquitin chains with this N15 on one acceptor ubiquitin and C13 on the other, and then we use these DUB enzymes through these DUB enzymes, and then they will cleave this bond, and when it cleaves this bond, then the intensity of the peak is going to drop with time. And we can do this experiment and look at the rate of the drop of the enzyme and monitor that to get the activity of this enzyme, which is the deubiquitinating enzyme. You could do that using the HSQC experiment as well. You could do that using the N15 side chain labeling experiment as well. So we did something very interesting here. We just expressed the DUB enzyme as a E. coli in the E. coli bacteria and used the extract. We didn't even purify the enzyme at all. We just used the extract, and we could see the effect of the DUB enzyme and the rate of the amount of the substrate, which is the K48 diubiquitin, decreased over time because it cleaved this bond, and we could measure the activity of the enzyme. So for example, now if you start thinking about it, you can, if you are interested in looking at the activity of the enzyme in sort of cellular conditions, you really don't want to purify it and do it in vitro conditions, you can just overexpress the DUB enzyme and then look at its activity. And if you want to design drugs or test drugs against it, you can throw those drugs in the same mix and look at the rate of the cleavage of this bond and measure the activity, whether DAB inhibitors are actually working or not. Just tell you that DAB inhibitors are a very important target for cancer therapies, because in many of these melanoma cancer cells, the amount of these DABs are overexpressed, and so once they are inhibited, it seems to give rise to some benefit. And now I would, one can also think about looking at this in cellular conditions, in cell conditions, using in-cell NMR and all of that. That's up for more discussions and experiments. So I will conclude here. So in general, we discussed 3 simple techniques to monitor ubiquination in real time by NMR. The temporal resolution actually can be used further by nonlinear NMR techniques. I don't know if it is going to be discussed, but there are several non-linear NMR techniques available, like non-uniform sampling and all of that, which can enhance the time resolution of these experiments. And the techniques that we discussed can be applied to any amine to amide post-translation modifications. Ubiquination is not the only one that changes the amine to amide bond on the lysine side chains in proteins. There are other modifications as well which change lysine side chains, like acetylation and all of that. And all of these post-translation modifications can be discussed. While we discussed only ubiquination today, but as I told you, it can be used to monitor any enzyme reactions. NMR can be used to monitor any enzyme reactions. The critical thing is that the substrate and the product resonances should not overlap, and there should be an internal control which will allow you to monitor the rate of the product formation. If you think that for some reason there are chances of overlap, then use some clever isotope editing techniques or isotope labeling to get rid of that overlap. There is a lot of discussion that you will hear over days about protein dynamics, and I just wanted to highlight that enzyme kinetics is also regulated by enzyme dynamics. For example, this very important case of adenine kinase. This adenine kinase is a very interesting molecule to look at enzyme kinetics and dynamics. And around 20 degree centigrade, this goes into 2 conformations where this lid kind of closes on the active site with the substrate molecule you see here, and the kinase becomes open in this conformation. So the catalytic rate in these conditions of this enzyme is around 261%. So the question is, what is regulating this catalytic rate? What changes in the protein can affect this catalytic rate? In this paper back in, okay, I don't have it here now, but it's around 2010, the group showed that actually this lid opening and closing has these rates, which is the closing is very fast and the opening is slower. And the opening rate is absolutely in sync with the catalytic rate. So effectively, it is regulated by the rate at which this loop opens up. And one can change temperature, and if you change temperature, the catalytic rate is going to change, but that also changes the rate of this opening. So essentially, you would see the active site of enzymes in many cases have loops that open and close, and the dynamics of those loops regulate the activity of these enzymes. So it is not only structure that is important, it is the dynamics of protein which is also important. I will not go into details of protein dynamics because I see that there are many classes that will come up or lectures that will come up about protein dynamics. So I will stop here, thanking the NMR facility, the X-ray facility, and some other collaborators, and I will be happy to take questions on monitoring enzyme kinetics by NMR. Any questions? Is crystal clear?
I would like to ask.
Sure.
I've seen, what is the size of donor and the acceptor protein?
They are both around 8 kilodaltons.
It's small. Is there a size limitation in these experiments?
Not really. I haven't shown data here today, but I mean the product essentially cannot be more than 30 kilodalton because of NMR related problems. If you are looking at the product as a protein thing, which is why we cannot really, for example, looked at polymeric chains or chains that are forming more. We have designed the system such that the product synthesis is going to stop at diubiquitin, but we are going to answer why a specific linkage is formed. If these polymeric chains you want to monitor beyond a certain range, which becomes difficult for NMR because of sensitivity issues, then it's better to use some other techniques, other biophysics techniques. But for some example, I mean there are many cases, some cases in our lab as well, where the product and the reactants are small molecules, and for that there is no size limitation now. So in those experiments, you have the product and the reactants in the NMR tube. The enzyme is low concentration. So generally these reactions enzyme is low concentration. So if you just take a proton 1D spectrum, for example, you would not see the enzyme, but you will see the rate of the reactants increasing, decreasing, and the product increasing. And you can do that in 1D proton experiments. If you have specific C13 label, then one can do C13 experiments as well. But if you use abundance, just natural abundance C13, then it becomes a little different because the experimental time increases and the temporal resolution then goes down. But for example, if there are small molecules, acetyl groups being formed or not formed, those reactions can be monitored within 5 seconds or so. There are several groups in the campus who are doing this, and they are getting good results.
And I'm also curious that why when you in N15 you check the proton in for 10 minutes, why you didn't get the other NH protons in that spectra? When did you get?
So the trick is that is it this one or the one
yes,
before?
N15H metal.
Oh yeah. So this labeling happens such that only the side chains are labeled, and on the side chain N15 epsilons are labeled in the lysine, the others are not. So you essentially get a lysine that is only labeled at the N15 epsilons, and if you use that in your minimal media, then only the lysine side chains are going to be labeled and nothing else. Now remember, lysine side chains are not observable. They are only observable only they become amide from amines.
Okay, I thought that the N15 is added in the media only, but now you're saying lysine is the only one which is N15.
It's a normal N9 media, but only added the
lysine.
Okay,
thank
and
you.
The other one was easy because there's N15 and C13,
Yes,
so you have to.
Thank you.
Only then you can see them, because the rest of the lysines here are still amines, and amines are not observable.
Thank you very much, sir. No, I don't have a question.
Okay, let me see. Because I can see the time of this reaction rate, basically 41 minutes, then 67.
Right.
So seems to be reaction is slow. Suppose because your experiment 10 minutes and you have further gone down to 5 minutes.
Well, I think we have plotted for the resolution thing. We have, to people to see the.
Yeah. That's okay. Maybe you can do for 1 minute also.
But the resolution in this experiments only went down to 3 minutes.
Yeah, 3 minutes. Then also is fine. But suppose there is a reaction, means some
It is
faster than say 5 minutes. Within 5 minutes, so 10 minutes it is completing.
Right.
Suppose, okay, at physiological temperature.
Right.
So there is an what will be your recommendation for alternative?
Well, I think for ubiquination it is difficult.
Okay.
For the other enzyme reactions, one can go down, the proton acquisition time can go down. But for these reactions also, there's something that's very interesting that we have to keep in mind. We have to reduce the dead time as well. So we need some hardwares attached. There are some research groups which has proposed hardwares attached, which we can attach to the NMR spectrometer, such that the dead time is reduced to 0.1 second, something like that. So it has the NMR tube as the sample, and there is a other capsule which has the this one, the reactant or something that activating. For example, in our case, it was ATP. So ATP will activate, otherwise it is nothing is going to happen.
Okay.
So, so you have that ATP as a syringe, and when you say, okay, go, then the syringe will drop the sample and it's done.
And other way which I can think is maybe diffusion edited. Maybe you can answer better. Suppose I have ubiquitin and that substrate donor and acceptor.
Right.
I started the this reaction and I started recording the two diffusion edits, two-dimensional. Okay,
Right.
and that completed 14 minutes or 15 minutes. That 2D can be completed because proton signal will be measured on that dosing. So can we do the same dosing experiments? Quantitative, I don't know whether quantitative estimation is reliable or not.
Yeah, that's what we have to test and see. Also, the difference, remember, is between 8 and 16. So
No,
we
but
should have a different, substantial difference between 8 and 16 to see a pattern in dosing that one can calibrate and get the intensity.
Means this separation between 1D and 2D strips.
Right. Size difference is only by 8 kilodaltons.
Means what one T is telling means another way will be like what you do in same thing use in the enzyme kinetics in the T1 dimension you detect the actually the reactant, and in T2 dimension the change part. So if you add it, so in the first step you have the signal T1 is coding for the initial state and T2 is coding for the second state.
Okay.
So that is like a photo reactions once when you have the pulse laser putting it inside and you detect the first part without irradiation in T1 dimension, and T2 dimension is only the irradiated part.
Right.
But it requires a very synchronous with the pulse sequence. That's another way out.
Synchronous with the
Pulse
syringe.
sequence. Yeah. And the second thing, Ranabir, which you showed in the last part, the dynamics and the kinetics. So this holds true when your kinetics is in comparable range as far as dynamics is concerned. So if your on rate of the product is really fast enough, is very, very large enough compared to the dynamics, so dynamics will not have any much effect on that, because any time the product is going, the on rate is so fast that it will see a state that it has to bind, and equilibrium will slowly start shifting on the other side.
Exactly. If the on rate of the substrate is very, very fast, then the loop is not.
The dynamics will not play much role.
So this we generally see with the whose size of the substrate is a slightly comparable amount, which makes this diffusion rate of the substrate into the active site slower, comparable to the loop dynamics.
Hi. To the comment that Dinesh made, we can slow down the kinetics and measure that, right, at low temperatures.
Yeah, you can do that, but then again biologists will be unhappy.
Okay.
This is not the real. If you want to compare against with something else, yeah.
Okay. I have two questions in the presentation.
Yeah.
One is your DUP kinetics. You measured nanomole range. Is
Yeah.
using NMR or?
Yeah, so the rate of this formation was in, this is actually not nanomolar, this is nanomoles. So nanomolar into volume.
Yeah.
Nanomoles.
Okay. And second thing, you are adding the ubiquitin onto your substrate.
Right.
It's one at a time, or you see 1, 2, 3, or in the same reaction mixture?
Yeah, so we have designed this with specific mutants of ubiquitin such that the reaction will stop entirely. I mean, this is essentially the reaction that we started off with, for example. Sorry for taking a little bit of time. So for example, this one is essentially stopping at diubiquitin. If we allowed it to go to triubiquitin, tetraubiquitin, then it will start forming more chains. Again, then the problem becomes is it becomes too large for NMR to again look at it.
Okay, thank you.
So if you have a question regarding how the processivity of the reaction is, how one enzyme versus another enzyme is able to build polymeric chains with time more, then this is not going to be the useful one. Have to think about something else.
Okay, sir, my question is that I have some kind of work like that only. So I have a very big enzyme and at the same time I have a very big substrate.
Okay.
So I thought to do the enzymatic assay and we thought to fluorescently label the substrate, but we were unsuccessful in that. So can I do enzymatic assay and do the NMR study?
Yeah, you can. So my first step is going to be take a NMR spectra of the substrate and the product. If you know if somebody has collected in the database something, just take a look at it and ensure that the resonance frequencies, that the peaks of them do not overlap. At least there should be one peak in the substrate that doesn't overlap with a peak in this one. Then you have a particular identifier that the product is forming, and then just use that.
Okay.
Yeah, I mean there are some groups in the lab, for example Ramaswamy Subramaniam, whose group was trying to measure the enzymatic rate of a particular enzyme, catalytic rate, and using fluorescence techniques they were not able to, or the rate was showing very different from what is probably due to tagging problems, but they could do it by NMR very effectively.
Okay, so individual of the substrate and individual of the enzyme, and then
And the product, sorry, product is important.
Okay.
If you have an NMR spectra of the product, NMR spectra of the substrate, match them and see that you have one at least individual peak of the product. Essentially you have, because the molecules are different. So you will have once at least resonance of the product that is very different from the rest of the resonances. And just use that peak to monitor.
So one more thing. So my substrate is tRNA. So can I get the spectra of tRNA?
Yeah, I mean one-dimensional spectra you will definitely get. At number of scans could be this one. Yeah, very flexible, very sharp peaks. Sure.
Just a quick question.
Yeah. Hey,
sir,
nice to see you.
Nice to see you too. So there are like 7 lysines you have, right? So can you correlate depending on the rate? Like one might be ubiquitinating very fast, one might be slow to the biological function.
Yeah, okay, so you are saying the different type of chains forming and they are related to form. So this, right now the biology itself is not clear.
Okay.
I mean, we have 7 lysines and these chains form different functions we know. But there are other complexities also. For example, there could be branch chains. A chain that started with lysine 48 and then went somewhere else. These are also observed. The question is, we can detect them by mass spectrometry, for example, but what is their function? To understand that we need a deeper understanding of which enzymes is actually doing it, or we can synthesize those chains with the substrate and then see what happens to the substrate. So these experiments are still, this synthesis of these chains, for example, if you want to do enzymatically or chemically and then do that on a substrate, it's been discussed and it's difficult, it's challenging. So people are working on it. Now, the one example that we have found very well is that, for example, certain proteins which have a DNA repair function. So they have a K63 type chain, lysine 63 type chain forming, which attributes to the DNA repair function. But again, after cells have DNA repair, this function has to be reduced down, right? It has to be attenuated, otherwise you cannot have DNA repair enzymes active all the time. So the attenuation is done by making this K48 type chains on top of this thing now, and those branch chains then lead the substrate to degradation, attenuating the function. This is one way of looking at different type of chains and their functions. But as chains are growing up in time, what is their function, we do not know. And also very interesting question that we are breaking our head into is, can we actually, if a substrate has multiple type chains formed, can we monitor all of them at the same time using NMR?
Thank you.
Well, it has to be some sidechain experiment. The question is how we will make the sidechains resolved, because the sidechain spectra just overlaps. Are there isotopes to do that? Okay. So that could be one way to think about it. And then that creates the difference, right?
Any more questions?
Yes. Sure.
Do the study with the mass spectrometry.
Yeah.
So what is the expected mass of this product? So because you have labeled and one is your unlabeled, so only one mass, 1 Dalton will be the extra mass in case, and that is one more confirmation that your product is formed.
Yes.
And it is on same RT. That can give you one more additional information.
Right. No, mass spectrometry techniques are very useful in studying these.
Yeah, because you have labeled one nitrogen, that will give you only 1 Dalton extra,
Right.
and the RT will be the same
Right.
retention.
The only thing is I don't know how to do this in real time.
Yeah, you
as
can
it
do
is
that
happening.
triple code MRM based technique. Okay.
We have to stop the reaction and then quench the reaction and
Yeah,
then
you have to quench.
That's possible.
Okay,
We were
th-
trying
means
to
this
think about-
study is to particular to establish the NMR based platform to do kinetic study,
Well-
not a part of another study where you want to do kinetics.
No, the question is, can we do the kinetic experiments in
Yeah,
real
okay.
time mode or not? And if you want to quench and do that, then these assays or mass spec, you could use anything.
Yeah. Okay.
Mass spec is actually extremely important to find out the type of chains formed and all of that. But the problem is we have to quench and do.
Thank you.
Thank you very much, sir.
But the other problem with mass spec is for every of these lysines, we need synthesized peptides which will act as the internal control. Otherwise they fly differently and you cannot calibrate the intensities properly. So for example in ubiquitin there are several lysines. For example I do a diazation and find out the peptides. For those peptides I need a synthesized version of this chain, and it's difficult. That kind of synthesis is costly.
Thank you, sir, for
Okay.
giving an insight how this versatile technique like NMR
Thank
can
you all.
be
Thank you
used
for your time.
to study enzyme kinetics and post-translation modification. Now, I would request Dr. Neil to present token of appreciation to
Thank
Dr.
you.
Das.
Excellent. Okay. Okay, I hope we have time. Did I finish on time? नहीं चलता है?
And we will assemble back at 12:15.
as Senior Principal Scientist at CSIR CCMB, Hyderabad, where his lab focuses on RNA binding proteins which play an important role in gene regulations. So without any further ado, I would like to invite to deliver the talk.
Can you hear me now? Yeah. So let me thank Neil and other organizers for this very kind introduction, kind invitation, and also for kind introduction. Some of it is this pointer is not working. The batteries are not there. Okay, thank you so much. Yeah, I'll actually come this way and then speak. Okay. हाँ दे दीजिए फिर वो फिर से लगाना पड़ेगा हाँ लगा दीजिए फिर से ये मुझे कंफ्यूजन होता है Okay, sorry for that bang. Protein dynamics is always like being banging the spins, so it's probably apt for what I'm going to speak to you today. Those of you who are interested in modern art and have been connoisseur of some of these paintings or have come across some of these paintings, this is a painting of Edward Degas, which I saw first time in the Metropolitan Museum of Arts in New York, Manhattan. And this is one of those paintings that Degas has made in multiple copies, and that, in my opinion, describes what proteins do inside a solution, as you can see, particularly for NMR spectroscopies. So this choreography for ballerinas that are going to dance over here, some of them, as you see, are oriented in a very uniform direction, whereas some others are wandering away. And nuclear spins behave something similar depending on certain of their properties. So this picture is somewhere tells us that how macroscopic world of ballerinas fits into the microscopic world of nuclear spins and their properties. And then this musician also reminds me of relaxations at lower frequencies, as you can see over there. Those of you who don't have background in proteins, I thought of putting this one small or two small slides over here. These are 20 natural amino acids that occur inside a protein, and they have very unique properties based on their side chains, which is shown here by the CH2OH, in this case is the serine. And as you can see over here, you have got a ring in histidine or in phenylalanine or in tryptophan or in tyrosine, whereas there are negatively charged residues, aspartate, glutamic acids and aspartic acid, like as you can see over here, whereas there are some positively charged side chains that you can see in histidine or in lysine. So this constitutes a diverse group of side chain based functional groups which makes the protein, and these side chains actually give very unique property to the protein surfaces. And in short time I am going to tell you how they are important. And these amino acids, as you all must have seen in the previous talks, or you must have studied during your BSc or MSc, that they have got this amino group which is connected to a quaternary carbon, which is connected to a carboxylic group, and this forms the backbone of the polypeptide chain. And as you can see over here, this is the backbone of the polypeptide chain. This is NH, this is C, the C over here, and the R group will expand from here. And in this particular schematic diagram, the R group has been silenced so that one can see the integrities of this particular structure. For example, one of the features that strikes your attention is this weak bond which we call hydrogen bond, which is formed by CO and NH, which is essentially required to stabilize the structure. So when these polypeptides come together, they essentially form this kind of a structure, which is called as a beta sheet, or an alpha helical structure, which I'm sure you have seen, right? What many of you might not have seen so far is that protein structures have a supremely vast network of folding, and the kind of shapes that they fold in is very, I would rather say that it can actually encompass more or less any possible combination that one can think of. And that is possible primarily because you have got so many such heterogroups like 20 amino acids that I have shown you. Whereas if you look at nucleic acids primarily, your nucleic acids are having only basically 4 bases, and then those 4 bases mainly fold, although I mean there are newer structures of nucleic acids that are coming up which are a little bit more complicated. People do see nucleic acids in a very classical way, looking at either a double-stranded helix or unstructured, chain of nucleic acids. Here is a paper which appeared in NSMB in 2015, and that paper describes how one can actually get the structures of proteins. And if you look at these structures, essentially if you look at this, these are nothing but alphabets in English. This A, B, C, D, E, F, G. Can you imagine all 8 is A to Z, And you take any other language, I'm sure you will find structures that will fulfill. Probably not in Hindi so far, but very soon, the way chi-EM structures are coming up, I'm sure some of you or many of you know about this new technique called the sky electron microscopy, which is solving structures at a larger scale. Now, this tells you, despite having, very common backbone platform, as you can see in this particular picture over here, and certain heterogeneity at the R group, one can, and of course the combinations of these, one can end up in making structures of proteins which forms shapes that you can just imagine, okay? And that's where we thought the structural biology would. But then what NMR spectroscopists like us found out that proteins are not just happy forming into A, or in this case J, or in this case O structures. They want to modulate these structures further, okay? So it's not that they're just happy with these shapes, because these shapes you get by the certain combinations of amino acids and this beta sheet or alpha helical structure. But these proteins inherently undergo some kind of changes. They remain, for example, this D will remain more or less like D, but it will show some change in this alpha helix or some change in this loop here, depending on how it is recognized in the substrate. So they are not just happy with having whatever structures that are there, about 100,000 or 200,000 in the database that you can see. Many of them are homologous to each other. But if you look at, there are several unique folds. And also there is one important thing that you must notice is that when you talk about unique fold, we talk about a fold that is not reported in the literature before. And for those of you who have studied X-ray crystallography and have also looked at NMR, you will find that the finding a probability of a new fold is more or less equal, and that's why completely new folds you will find discovered by NMR and X-ray crystallography more or less equally. Although X-ray crystallography is a very old technique, relatively old technique than NMR. Okay, so these are the structures, and these structures adopt certain changes. Okay, this is complex structure for that matter. There is an alpha helix that you can see over here. There are certain structures in which this alpha helix was sitting in the core here, and there was debate on this particular structure. Series of PNAS, Nature, Science papers, and people are still probably, the issue probably is not still settled. Okay, so why do you need to look at the structures? The point is you need to look at the structures because structures define the function of a molecule. And as you might have seen from the previous speakers, structures are also required for you to target. When you look at, particularly you are in the drug discovery platform here today, so what happens when you take a drug? Drug doesn't have any miraculous thing, right? The drug goes and shuts down certain pathways, which essentially means that the molecule in the drug, the small molecule in the drug that you are taking, has to go and block something or initiate something or trigger something, either positive regulation or negative regulation, depending on what it is. If you look at just subset of proteins, okay, this is a subset of proteins. These are called as G protein coupled receptors. I don't know how many of you know about GPCRs. I'm sure some of you have heard about GPCRs. GPCRs are essentially membrane encoded protein. They are bound in the membrane. They have this transmembrane domain. They have the domain that is in the cytosol, and there is a domain that is extracellular domain. So essentially this remains as a sandwich, okay, on the length of a membrane. All of you know that our cells have a membrane, right? This we call as a plasma membrane. This is formed by some lipids, and those lipids hold these GPCRs, and GPCRs anchors inside that. Part of the GPCR is outside, part of the GPCR is inside. So one of the structures that I showed you, was GPCR in that case. And GPCRs, why I showed these GPCRs to you is because GPCR, if you look at the GPCR pharmacogenomics, essentially until very recently we could not solve the structures of GPCR because they were membrane encoded. And because of that, both crystallographers and NMR spectroscopists, only very recently Brian Kobilka and others have solved some GPCR structures for which they got a Nobel Prize recently. And these GPCRs are receptors, they are sensors. Their extracellular region actually senses something. So when we take a tablet, the tablet goes and sits to that extracellular domain. And that's how there's some translation of the signal right from here, the extracellular part, to the intercellular part. And that's how the biology or the chemistry of a cell begins. So this is a very complex molecule, and there are many such receptors. So far 196 receptor ligand complexes have been determined. Some of the slides I didn't give references, that's just because I was in a little hurry, but that doesn't mean that this is our work. This is somebody else's work, we have picked it up. And when you look at the GPCRs, there are more than, for example, if you look at the genotypes of the GPCRs, there are close to 1,000 genome projects that have identified 2,500 individuals. And they found out that in these people there are mutations, there are significant number of mutations in the GPCR gene alone, which leads to some missense or loss of function or copy number variation and stuff like that. There are already 475 drugs that have been approved by FDA, and GPCR drug target markets to 108 trial GPCR targets are in the process right now. If you look at the number of non-targeted GPCRs versus targeted GPCR, approved GPCRs, you can see that there are still, and I'm sure some of these non-targeted GPCRs will essentially be coming into this approved category over a period of time when they realize where the interactions are happening. You go back and this is also in terms of monetary funds, it is essentially increasing. A large number of shares have been increasing for this particular type of drug. I'm just showing you a tip of iceberg. When I talked about a lot of proteins, I'm taking a subclass of protein to show you how important they are in drug targeting. And that's why it is important for us to know the structure, because structure can define the drug discovery program. And as I told you, structure is not just structure. When we talk about structure, we talk about structure coupled with the dynamics. Okay. So I will tell you one simple example here, how the protein structure is coupled to the function. We take an example of an enzyme, and we all know the enzymes recognize substrates and does some catalysis, does some reaction on it. Let's say this particular enzyme has a pocket here, which is tailored to fit a ligand with shape A. Now ligand A goes and sits here, and the enzyme acts and breaks down the ligand A into these two halves, right? In this shape recognition process, ligand B kind of shapes cannot go and fit there, or they fit very loosely so that the enzyme cannot act. And this is at the backbone of all enzyme chemistry. I'm sure Rana and others would have already told you about them. Okay. So the question that remained in front of biologists was, how do you go from this state to this state? What are the intermediate states in between? And are those intermediate states any important? And can we modulate those intermediate steps? And if you can modulate those intermediate steps, then effectively we can have a more potent drug, right? For example, if you are taking one particular tablet for a particular disease, and if you can moderate the dose in certain way, depending on the individual, depending on the mutations in the individual, one can actually get a remedy, or the patient suffers less in the whole process. So there are two major doctrines that have come in the mid 70s, or I would rather say early 80s. That was about the induced fit model, in which what happens is substrate A goes to the enzyme. Now in the presence of substrate A, the enzyme changes its shape a little bit, and by changing that shape, you have actually A fits into that groove, and the moment A fits into the groove, enzyme acts on it. That is called as an induced fit. Whereas in the recent times, maybe I would rather say last 20 years or something, people have come up with additional, of course conformational selection idea was there for quite some time, but there were no direct proofs for it. So other concept that has come up is the conformational selection, which tells that A doesn't really induce this. You can see there's a little mismatch here, okay? If you can see my pointer over here, A fitting into that enzyme cavity is not perfect. So to fit A perfectly over here, so basically there is still A would not be fitting here if enzyme doesn't move, this part of the enzyme doesn't move out, right? So to move that enzyme out, enzyme has to sample some conformational changes. So the doctrine was that this conformation, enzyme already has embedded information in it, so that it can open up itself, absorb A and cleaves it. And that's the debate that goes on in the field for probably even today. Probably because each enzyme acts differently and probably each enzyme has its own way of handling things, okay? So the idea is that to identify this ligand binding pocket over here, your original enzyme is like this. In the presence of ligand, the enzyme closes, that is called as induced fit. The conformational selection says that this enzyme has inherent property to go and close on its own. The moment it's in the closed conformation, if the ligand comes, it goes and binds. There was a very interesting paper in this biochemistry in about 10 years back, which looked at this protein MBP. This is a maltose binding protein, and that has got a nice groove over here, as you can see here. It has a similar groove like this, and then this groove opens up in the open conformation. People could crystallize this. This was NMR crystallography and MD simulations, and these were good old days. That's why they published in biochemistry. In today's world, they would have tried for eLife or something, flashy journal. So they have found out from the MD simulations that that groove automatically semi-closes. Okay, there is no ligand here. So essentially, instead of putting a complete induced fit, that means if your ligand has to bind, you open up, your structure is open, you close it, but you don't close fully. You just come closer to the closed state, and at that stage, the ligand now induces conformational changes, and that's what they have shown in their paper by doing set of MD simulations. And you can see that the ligand is actually sitting here very nicely. And they have shown there is some balancing interface that actually has to move away from this side over here, which is very close in this case, opens up little bit here and opens up further over here. And the allosteric changes that are occurring because of these changes allow the ligand to go and sit in there. So they named it as a mixed model, that says that both induced fit and conformational selection are occurring at the same time. So essentially that tells you that these molecules have structure and they have certain motion, certain dynamics. So we will try to understand what they are. Before we get in there, I want to bring you to a very basic 10th class chemistry lessons that all of us must have studied. And this is a nice quote I found out very recently from a very recent paper by a friend of ours, Hari. He wrote nicely, this is the beginning of his paper. He says that much like sculptor transforms stone into a structure or masterpiece using the artistic tools, medicinal chemists like you transform a molecule into a drug, into clinical settings using the laws of thermodynamics. And that's very correct actually. This is the opening sentence in JB&MR. I really liked the way he talks about. Now, we'll try to understand some of these processes, and since I didn't know much of the background of the student, I tried to do it with as little equations as possible. So when we talk about molecules, molecules have various properties at the room temperature. Of course, as you all know, that absolute zero, the molecular motions and other things freezes, but then there's nothing like absolute zero here. So you go around here and then your molecules diffuse in direction. There are certain vibrations in the molecule. Molecules also collide with the wall. And this is essentially, the molecules also rotate. So for example, this particular molecule rotates along this line. At high temperatures, these motions are faster. They are more pronounced compared to a low temperature. Now if we look at the container, the water molecule inside this bottle, you can see that this process is actually happening right now. And if you focus on one of the red balls or something, you can see this is, sometimes it is moving very slowly, sometimes it is moving very fast, depending on how it collides with either with the wall or with its neighboring molecule. So this tells you that these are the processes. This is a very fundamental thing of kinetic theory of gases. These are the fundamental processes that are happening in any liquid at any given point in time. Okay, and this is what part of this is what we cache in the NMR spectroscopy. Now, one of the things is that if you look at the Maxwell-Boltzmann distribution of molecular speed of the ideal gases, of course, I have taken this from the gases. You can very clearly see that you have got this kind of a shape. This is of course an oxygen molecule and you have got as if you increase the temperature in this particular case, at low temperature your velocities are low, at high temperature your velocities are very high, and there is something called as a very tiny fraction of these molecules have this VRMS, which is significantly larger, and that's one of the reasons why we don't have the gases like hydrogen and helium in our atmosphere, and we are flooded with things like oxygen and nitrogen, because the hydrogen and helium requires less VRMS to escape Earth's atmosphere. Okay, so when people were studying these things, what advantage dynamics gives us? So here is one of the first photographers who captured the motions, Edward Mabridge, he was from California. So one of the questions that were there in those days was horses run so fast that you can't really figure out whether horses can lift all the 4 legs at a time. And Edward tried to accept the challenge, and he started taking the photographs at a much faster frame rates, whatever was capable. Those are the glass plates, not unlike today's cameras that all of us have. Okay, and those glass plates, he took series of these things, and then he overlaid, and he could get something like this. Again, there was never a conclusive proof that it could it could lift all 4 legs at a time or not. Okay. What he did excellently was he actually started playing them really fast, and when he started playing them really fast, he could actually snapshot some of those places where the horse actually is lifting things. So basically, when you talk about dynamics, you need, in addition to you compromise resolution versus information, because each of these images is far more informative compared to the information where the horse legs are in motion. Okay. So this is one way where when we have too much of dynamics, you actually lose some more information in terms of structure in proteins, and you can see how that happens. So let me take you to very fundamental motions, okay? And these are some of these things called as translation motions. Most of these figures are taken up from a book called as Spin Dynamics by Malcolm Levitt, and I encourage you to read it. It's one of the very fantastic books to understand basics of NMR spectroscopy. So what happens here is when we talk about molecule as a particle, and we have shown this in one of those previous things, how the ideal gases behave, you have 2 processes that are happening. One is of course a diffusion, and diffusion occurs as I have shown you in that particular image randomly, and it hits, the molecules hit each other or hits the wall of the container, and they diffuse inside the system. Whereas there is something called as a flow, and flow is directional. It could happen in NMR spectroscopy due to gradients for that matter, or it could happen because of convection currents into your tube. It could happen because of many things, and then when you have flow, you have directionality. And when you have flows, then you have essentially molecules that are essentially diffusing on this axis over here. And if you see here, the molecular motion is far simplified in this particular direction compared to this. And this, because in this direction the movement is restricted because of the wall of the container. So this is called as anisotropic movement of molecule. Okay. Now this is one type of motion, what we call translation motion. The other thing is rotational motion, and this rotational motion, your molecule actually rotates along its own axis. And when you rotate your molecule along its own axis, you can see, and for example, if you take a small molecule, it takes lesser time to make one complete rotation compared to a bulkier molecule. And it is very natural that if you have a big ball and if you rotate it fully, it takes more time and energy to make one complete circle. In biology or in NMR spectroscopy, such rotations can be actually measured, and this is measured as something called as rotational correlation time, called as tau c. Tau c essentially is the time that molecule takes to rotate by 1 radian. And probably I'll tell you a little later why that is important, and how these motions actually help us in getting some of the subtle molecular features from a thing. So some of these are the motions that are considering at the molecular level. Like consider each molecule, and these are some of these motions where you have a translation diffusion. Yeah, you have a question? Yes, please. Yeah. Yeah. Okay. Yes, diffusion is Brownian motion. Yes.
In that diffusion was defined as a process of movement of
gradient, concentration gradient.
Yes.
So that is a type of, in that sense we can say diffusion is a flow.
If you have a concentration gradient, yes, of course. But here I am talking about in a completely isotropic, a complete homogeneous solution, particles will diffuse. Suppose we start, in these kind of auditoriums, then before we start giving our talks or coming assembly here, the people who maintain these auditoriums, they come and spray some perfumes or air fresheners, because these rooms are closed and then there's some suffocating smell or something. So they don't spray in every chair. They just spray 2-3 corners, and those particles diffuse in the room for a period of time. By the time people start coming in, they diffuse, right?
Yeah.
So there is no directionality there. They diffuse all around, right? Like if you take, it's a simple thing, you take chalk dust or white chalk dust, even if you have ink pen, you take a drop of ink and put it into this water bottle. You see, because of the viscosity of ink is slightly different, initially it will start settling down slowly. And it will create some kind of a shape, it will create its own some kind of a structure around the water. But as it starts going down, eventually the water will start breaking inside, and then you will have a very homogeneous color. We do, I'm sure you play Holi, and you can do this exercise next time you are playing Holi. You can have, if you take solid powder and pour water into it, and don't mix it well, and start using that color as it is, very little particles will diffuse up, and the initial part of the color that you will be taking will be very less dense, but as you are slowly coming down, it will become more dense. Same thing happens with the canned bottle juices. You have to shake it. Because of the particle size is higher, they won't settle down. That's we decant things, right, in the laboratories many times. So something like that. But when the particles are of identical size and identical shapes, they essentially don't remain there. They have their internal energy, internal kinetic energy that allows them to diffuse inside the phase.
So I can say diffusion is diffusion is due to Brownian motion.
Yes, diffusion is because of Brownian motion.
Okay,
Yeah. Flow comes because of directionality. Like for example, here there is a AC going on, right? Then there some flow comes. Then if you spray in front of the AC, then that flow will take it rather than the room will getting saturated, right? So in NMR, flow has certain specific meaning. I'm not getting into the detail. There's something called as a gradient axis in the NMR, and one can actually use them to create a flow of that. Or if you increase the current, there's a convection sets in, and that creates a flow inside the tube. Or if you have high salt inside the NMR tube, that creates some kind of electric field around it, and that creates some additional artifacts, okay, into the spectrum. Okay. Any more questions? It's good that you interrupted me in between. Please, this is something I want to tell people, that this is only to make me understand what dynamics is, and a little bit for you people to understand also. So if you have any questions, please stop and interrupt and ask questions, okay? Oh, anyway, I didn't describe this part of the slide, but this is essentially what I mentioned some time back about various motions. This rotation, this is at the atomic level, so I will cover it in the next thing. So when I talk about here in this particular things, I'm looking at each molecule as a particle. But each particle also in proteins is composed of atoms. And when you look at those atoms, these atoms are like this. For example, here there is a CH group or methyl group. Okay, in the previous one, it is shown very nicely here. Okay, this is the rotation of methane. So you have methane has this axis, so you can actually have rotation of this methane going on like this. So if you come here, this is a C connected to a hydrogen, and this H actually rotates. Even the NH on the protein backbone, if you have this polypeptide chain, you have got NH, so N and H makes some kind of a cone. Subsequent slides I will tell you how much it can tilt and how much it doesn't tilt, and what happens when it is very rigid, what happens when it is very mobile. Okay, but these motions are inherent, and these motions can be only frozen at absolute zero, which is not going to be realistic for many of these molecules ever. Okay, so this is a motion that happens all around, and this motion essentially is a rapid oscillation around this axis of CH, and this happens primarily in the picoseconds or sometimes nanoseconds. These are called as vibrational motions. Okay, this is very important for you to notice this, and this happens for most of the XH kind of molecule. The other motion is called as rotation motion. This is for the methyl group here. For example, on this carbon there is an axis of symmetry here that is going on, and there are 3 hydrogens on all 3 sides, essentially like this, Consider this is a carbon and you have got 3 hydrogens on this side. So they rotate like this. This is called as a rotational motion. So basically it rotates around this 3-fold axis that I am showing here. These are also very fast motions, and that's one of the reasons why you get very sharp, nice signals of the methyl groups most of the time in the NMR. Sometimes you have broad methyl group signal, then you know that these methyl groups are involved in some kind of dynamics. That's a very hallmark feature of it. So these are the 2 major motions that are occurring at a very fast time scale. In NMR, when we say fast time scales, we talk about picoseconds, nanoseconds. Okay. When we say slow time scale, we talk about millisecond to second. Nothing after second is there. Okay. I mean, it's not like hour or year or light year. Light year is, by the way, not really a unit for time, but this is essentially occurring at very, very fast time scales, whereas the other motions are occurring at relatively slow time scales. And those relatively slow time scales are chemical exchange. For example, this particular molecule, this hydrogen over here, gets chemically reoriented, and you have got this particular new structure forming, and now this is in equilibrium. Okay, so these 2 molecules exist at the same time, and they are exchanging between 2. But these are 2 different chemical entities, and because of that, that's called as chemical exchange. There are other exchange processes that happen. There's something called as a conformational exchange, where you have got an orientation around this N here, and there's a rotation around this bond that makes these CH3 groups rotate around that nitrogen, and that's called as conformational exchange. I have a beautiful movie. This is based on some of the work of one of our colleague in NMR spectroscopy, Hashim al-Hashimi. He has been looking at some of these fancy base pairs. In the DNA and RNA, there is something called as ATGC, or in case of RNA, it's AUGC, and they form base pairs. And what we have learned is that A forms a base pair with T and G forms with C, right? But then there are something called as mismatches or oval mismatch pairs or something. So he studies them, and he uses a lot of intense NMR dynamics to understand how the structures alter. So here's a movie. I'm just going to show you. This is the GT wobble. This is DG and DT means this is from the DNA. So it forms all kind of structures over here. You can see this, and you can actually see all of those structures. He shows it using a technique called as R1 row, and you can see that there are these significant transitions that are occurring over here in the molecule. There's also water that has catalyzed this. I'll play this movie once again for you so that you can have a feeling how these motions occur, how these conformational changes occur. And they play a very important role in terms of fluidity of double-stranded DNA. We think that double-stranded DNA is very rigid. Double-stranded DNA is not as rigid as double-stranded RNA. Double-stranded DNA is far more fluid. It can open up, it can close, and that's probably one of the reasons why he claims that DNA has been chosen as a molecule of life or evolution compared to RNA, because we believe that the world began with RNA, right? Okay, so now I showed you different type of motions here. I showed you different type of conformational or chemical exchanges. Now I also told you about some time scales, but it was probably not very clear. So I just quickly give you a brush of the ideas. We talk about something like 10 to the power of minus 12 seconds to 10 to the power of 0 seconds. There's like 1 second here in this particular point, and these are the NMR time scales. So these are the interactions these are the NMR experiments that you can actually use to understand the motions that are occurring all the way from extremely fast time scales to slow time scales. Okay, and between fast and slow, we have something called as intermediate over here, and we call these motions as intermediate motions. And these intermediate motions one can actually study using some of these techniques over here. In a very short time, I'm going to tell you very little about these techniques, because the kind of descriptions that you come across for these experiments in the research papers or textbook is much more complicated, and that requires a little bit more refined ideas about NMR spectroscopy, which I'm sure you all can gather. Now, in addition to this, I want to show you here something called as the free energy landscape. When you know that the protein folds, and I think Ranabir was talking about protein folding, or when protein folds, we all know about a protein folding landscape. So you have got a protein folding landscape, you have got a energy minima, what is called as a local minima, and then these many local minimas eventually come together, and there is something called as a global minima. And global minima is the minimum possible energy that the molecule can have, because it is much more stable. Like molecules also want to be more stable. So the concept was that the global minima would go like this. But what NMR spectroscopy, particularly from the lab of Louis K, it has come out that global minima is not just one global minima, not just one single conformation. What Louis K has told here is that the global minima has multiple such transition states, and each of these things actually there is he is talking about two different conformations here, population A and population B in this particular case. But even in A, you can see that there are a series of picosecond motions or nanosecond timescale motions that are actually converting your molecule from one particular minima of conformational coordinate to another conformational coordinate. So even molecule on its own undergoes these kind of transitions here. Okay. So on his there is a molecule called as ubiquitin. It's a very well studied molecule by NMR spectroscopists, primarily because ubiquitin is into doing its biology. It has a certain role in biology. It has also helped NMR spectroscopists to build a set of experiments. So ubiquitin is considered as a rock, a solid rock kind of a molecule. Okay. Louis K's lab has actually shown that even ubiquitin has this kind of motions. So that means that these are so ubiquitous in a way. If I have to use that word again. Okay. So what happens when you are having these kind of conformational changes? How does an NMR spectrum look like? So let me just show you one classic example here. This is a very early paper in 60s or something, where there are two systems A and B which are undergoing equilibrium population, and this is an NMR spectrum, okay, a single line. In this particular case, this is recorded at temperature of 193 degrees Celsius, very high temperature. But as the authors of this particular paper started reducing the temperature, this signal started becoming broad, and further broad and further broad, and then it reached to a point where you can actually start seeing the two peaks. And when they reduced it further from 139 to 136, you actually have developed these two nice peaks here, envelopes, and now it has more or less completely shifted into two populations here. Although they are not still baseline separated, that's why you have some exchange rate going on. But the moment in the next case at 100 degrees Celsius, they are more or less reached to the baseline separation, so there is no second population. Now you can actually detect both the populations here, and you further lower it to 50 degrees, you can actually see two signals. What does this mean? This essentially means that at lower temperatures, so as I showed you in the very second or third slide, that you can modulate the exchange processes using the temperature. Here in this particular case, you have got at a very low temperature, you have got two populations or two conformations A and B completely separated. But as you increase the temperature, you start having an exchange between those two conformations, and those exchanges in this particular case, as you can see, essentially coalesce somewhere around here at this particular temperature, and then eventually you can't distinguish between the two populations. Now whatever one single population that you are seeing over here is a mixture of these two states, right? And this is how the time scales matter to an NMR spectroscopist. When you want to identify both the populations, you need to play around with certain exchange parameters so that you can actually distinguish the information coming from them. Okay. This is also manifested in this particular saying. I mean, you can see this. I mean, this is the confirmation. Okay, this is gone again, I think. Maybe I'll use. Okay, this battery is not working, so I will try to. No issues. So my mouse is also not working. I can't show you the mouse there. Okay, so coming here, that we have got this, in this particular middle panel that you see over here, you can see that the central rod that you can see, which is a crossover return, that's where the line shape becomes really broad. And you are actually going from these two identical states of the identical population versus in the fast time scale motions. You get a single conformation here. So you can see the similar transition actually has been plotted over here as a function of time scale now. So here we had only information about the exchange rate. In this case, we have information also about the time scales now. Okay. So what happens when we try to plot them as a function of both? You can see these transitions very clearly in this case. Okay. Now this is for the symmetric exchange processes. Now if you have asymmetric exchange processes, where population A is low to begin with, and population B is high, then you can essentially see when you are in the slow time scale regime, you can actually see both the signals on the top panel. Where I wrote slow exchange, you can see a very small peak coming from A and a big peak coming from B. But then when you coalesce these two, instead of having a signal coming at the center of the frequency coming from both away from both the signals or in the middle of both the signals, you actually get it shifted towards the higher population. So some of these features actually give you information on the exchange rate, some of these features give you information about the populations, and some of these things can give you information about the time scales, and that's where it becomes important. Okay, so I have to now wait for his stick, because these are some of the relaxation, NMR relaxation processes. Have you studied T1, T2 NOE before by previous speakers? Have they taught you about that? Have somebody mentioned about it? T1 and T2? Okay. So T1 and T2 is relaxation methods in NMR and relaxation processes in NMR. When you have equilibrium magnetization, you perturb it by application of a 90-degree pulse. You get into the rotating frame, and from there the time it takes to diffuse into the same plane versus the time it requires to come back to the equilibrium magnetization. These are the time constants that they define. But behind the time constants are these mechanisms or these processes. They actually contribute to those T1 and T2s. Like for example, if the very first image that you see at the end over there, where I'm talking about dipoles, you can see there's a center, there's a atom, and next to that there's another atom, and that other atom is also part of the same fringe field of the first atom. You can see that as the molecule rotates, their orientation with respect to change, and that causes something like a change in the local dipolar field in the very first panel. In the second panel, there is a modulation because modulation of the chemical shift, because of the interaction between the two fields over here. The third process, what you can call, is the wobbling around the local field. So the local fields that you have around your atoms, they contribute to something called as additional motions over there. Now, if you take the sum of both external fields and the fluctuating transverse field, which we have seen in this number 3 case, you can actually see as a sum over here as a combination of the two. Thank you so much, boss. It was so difficult to explain this slide without this. Okay. So I was talking about the local dipolar fields. So here's an atom, the other atom is here, molecule rotates, you have got a different orientation now. Previously it was orienting towards each other like this. Now they are orienting away or slightly tilted at a different angle, and that causes something called as relaxation. Some relaxation mechanism, there is a field that is induced essentially by these two axes because of the rotation of molecule, and then there is something called as wobbling around the local field over here. Now if you have these, if you take the sum of it and try to understand how the field is fluctuating, this Bx is fluctuating as a function of time, you can see that this is actually fluctuating in a very random way, and if you take the average of this is going to be 0 more or less, okay, at any point in time. However, if you take the square of this, essentially it will give you some kind of a rotation, and then some kind of rotation in the positive side only, and that's why we have got this b square x, and that motion now you can actually study using something called as an autocorrelation function, and this autocorrelation function gives you a plot as a function of time, t or tau in this particular case. You can see that this autocorrelation function actually decays as exponential decay, and if you plot this autocorrelation function or on a long time interval, what essentially you find is that you have got at this particular time tau where you are looking, and then at certain time where there is some kind of inversion of the sign of this field, you have something called as a tau here, and this tau is more or less correlates to your rotational correlation time. Okay, so that's why the rotational correlation time comes up here. There is an inversion in the fluctuation in the field for this particular case. Okay, so these are various relaxation processes at the back of your T1 and T2. Now it is working. Okay, thank you so much. Okay, so how does T1 and T2 occur? So if you take the log of T1 and T2 as a function of correlation time, This is for various molecules that I'll come back to these numbers over here. What is this S square is here? You see that the T1 essentially comes all the way down, and these are the correlation times, very, very small correlation times. And these are not really called as only tau c, as I said, I told you that rotational correlation time tau c is a time that a molecule takes to rotate by 1 radian, right? You remember? And that's the time. That's the correlation time. Suppose my protein has 9 nanoseconds as a rotational correlation time. That essentially means that my protein takes 9 nanoseconds to rotate on its own axis. 9 nanoseconds to do 1 radian rotation. So here in this particular case, this particular region over here has a very low tau c, which essentially can be only fulfilled by very small molecules like methane or something like that. When you come to proteins, this is the grey bars are the ones which are really what is more relevant for proteins going from 10 kilodalton to 60 kilodalton. And here you can see that the T1 essentially increases as you increase the correlation time, but T2 decreases as your molecular size increases. Okay, and if you look at some of the internal motions over here that are occurring at few picoseconds to few tens of picoseconds over here, there is a drop in the NOE value. NOE is essentially a manifestation of a dipole relaxation. Okay, so all of these relaxation times T1, T2 and NOE one can essentially study as a function of correlation function, the 2 times the transformation of correlation function, which is called a spectral density function here. So this is a 0th order spectral density function, which essentially tells you that the molecule has a lot of internal motions. So if you take a larger molecule, you often the relaxation processes can be purely described by J0. Whereas if the molecule is of relatively smaller size, when I say relatively smaller size, I am talking about 10, 20, 30 kilodalton protein, you actually can describe the motions inside the molecule, all the features that we just some time back talked about, by these essentially 0th order spectral density function and omega n and omega h, and of course the combination of omega h and omega n. Okay, so this is one simpler approach to look at the molecular motions after you have collected the T1, T2 and NOE data for your protein. Okay, but that doesn't mean that we get all kind of information. So basically it only tells you that there are certain motions occurring at this particular time scale, but you can't get the details of those information. For that one needs to go back and do the more sophisticated experiments. I'll give you a simple example on how you identify motions inside a protein. I talked about here about this NOE, and I told about certain motions that are occurring at various time scales here. Here is also an NOE experiment. It is called as 15N1H heteronuclear NOE. This is on a protein that we have collected. This is almost I would say 42 kilodalton protein or something. We couldn't collect it ourselves fully because we have to actually take one domain here, one functional domain here, and another functional domain over here. And what we find here is very interestingly, so NOE is a very simple manifestation of identifying fast motions. Okay, if you have a protein and you want to understand whether the protein has a lot of functionally relevant fast time scale motions occurring in nanosecond time scales or something, very fast motions, one can actually do this 15N1H heteronuclear NOE. You can see here that for most of the rigid region where there is a structure, you have a very nice heteroE of 0.8 or something. Whereas when there is a linker between the two helix, a little longer linker, it drops down to something like 0.3 or 0.35 or so. And as you go back to the structured region over here, you again have a more positive NOE. But there is a large unstructured region over in this particular case, and you have a negative NOE here. But even in this negative NOE, you can actually see that there is a pattern, and this pattern tells you that these parts have propensity to form more structures compared to these parts here. So these parts are supremely fast mobile. Now what does it actually mean physically when you talk about it? I told you about rotation along the NH bond vector or something, right? You have got N fixed over here and H, which is a NH bond vector. So NH bond vector actually tilts and rotates like this. So this motion is very fast, and this whole set of amino acids, they are undergoing this kind of motion. Then you see this kind of heteronomy over here. Whereas these NHs are very rigid and they cannot move, or if they move slowly, or their tilt is not that much over the angle. You actually see something like this rigid part. Even between the pictures, you can see that a drop in the heteronomy in this particular region over here, and that actually indicates you that these are unstructured regions over there or something. Is this clear to you? This is one experiment, and you can actually get a lot of information from this one experiment about the protein dynamics. Yes. Yes. Small peptides also you can do head anyway, but small peptides essentially you will end up in having, depending on the flexibility of the peptide, of course, you end up having a lot more flexibility. But if you have belief that your peptide is rigid for some reason, for example, it's a cyclic peptide, then it has got a lot of restrictions, right? In that case, you can sit down. Yeah.
If we know that my protein is rigid enough, then I can, but for some time at least.
Yeah. See, for example, you have a hunch that, for example, you have got a long loop here. In this particular case, we have a long loop in this region. We want to know whether that loop is, rigid or flexible. And one can actually do this very simple experiment to know how flexible this is. So here you can see that this loop is a very small loop, has an heteronomy dropping almost to 0.6 or something, or 0.55 or something. Whereas a very long loop here has 0.35. It's not that flexible. So essentially this part is much more flexible compared to its counterpart, whereas this part is much more rigid compared to the kind of amino acids that are embedded in here.
Okay.
So you can get a very primitive information about how your molecule is, what regions of your protein are flexible, what regions of your proteins are dynamic. But this is again at a very fast time scale. This does not give any information whatsoever to you about the motions that are occurring in millisecond or microsecond time scale. This is only at the nanosecond. So if you do this, I know that this part. So for example, I had a hunch that there would be some structural features in here. I could not see any structural features in here because I know that heteronomy is so negative in this case that there won't be any structure because they're so flexible already that this region is like intrinsically disordered region which will go all around, and that you can also make out from the many other tools, like for example just simple look at the nitrogen line weight, or you can look at the spectral dispersion in the nitrogen dimension or the proton dimension in the HSQC and stuff like that. But in addition to that, or probably I think rather say more than that, this particular information that I have gathered here in our study was very helpful for us to understand that this particular region is extremely flexible. What was funny was we could not assign this region, and then we found out that there is a hidden domain over here which you could not predict neither bioinformatically nor structurally, and that has time is up. Oh man, not 2.5. Okay, so I will go quickly. So here I have another way where I can show you about the R2 and R1, which is nothing but the inverse of T1 and T2. And in this particular case, you can see that we have got a very flexible. We have got a very flexible part over here. We have got a relatively rigid part over here. Again, a linker, and another flexible part here. Now you can see that there is this heterogeneity in this particular region over here and here. So here the average R2 by R1, if I am taking, is about 15, and here average R2 by R1 here is about 25. That information is missing in the heteronomy. In heteronomy, both are identical. You see this? This answers your question, right? In heteronomy, both the domains have exactly identical information. So at fast time scale, nanosecond time scale, both domains are behaving identical. But when you look at the slow time scales, this domain has higher R2 by R1, which is 25, approximately 25, okay, 20.94, and this one has 12.91, right? Okay. Now what we did, of course, to get rid of that particular problem, we eliminated this N-terminal region over here, which was extremely flexible, which was flexible on the faster time scale, and then we now get both of them with the equal thing. I will not get into the biology of this. So to analyze these things, in addition to that, you can actually use, I talked about spectral densities, right? So there is an approach called as reduced spectral densities. And reduced spectral density is where you plot the J omega n versus J0 and find out how your molecule is. In this particular case, this particular domain is extremely rigid. You can see all of these things coming together, and J0 is very small. Whereas in this particular case, there are regions which are extremely flexible and they encompass over here. But when I concatenate these two domains here, the first domain which was flexible also became, which was rigid also became flexible in this particular case. Okay. So the other approach that one can study is something called as a model-free formalism that was established by Lipari-Zabur in 1980 and then extended by Clore in 1990. And they consider certain models, like model 1 where you have what this order parameter, a square of the order parameter where you can define the motions, and then there is a chemical exchange contribution in model 3, and then there is an extended Lipari-Zabur where you have got two side jumps, which is a slow motion, and the fast motion that is described by the motion on the cone. Okay, one can actually study proteins in this Lipari-Szabo model, and you can get the sense of order parameter. Order parameter is the square of the order parameter when it is approaching 1. Essentially, it is very rigid. When it is approaching towards 0, it is very flexible. Now, how does, okay, I will skip this particular part. This is, so there are some internal motions that you could find out in this particular case upon ligand binding. Okay, so, okay. I think this is very important for you to know, so I'll just go over this particular slide. I told you about relatively fast motion so far, nanosecond, picosecond. And there's something called as millisecond, microsecond timescale motions that we talked in the very beginning. Now, what happens in those cases? Suppose I have a polypeptide chain A here, and that polypeptide chain A is going into another conformation in polypeptide chain B over here. So, if you see here, I have got this particular nuclei spinning like this as in the population get exchanged, and it goes from this state to this state here, okay. And that motion happens in millisecond to microsecond time scale. There is an experiment called a CPMG. This is Carr, Purcell, Maubam and Gill sequence, very innovative sequence. And if you do this exchange at a very small spin lock field, you get a R2 effective of this size. Okay. Now you increase the spin lock field. Now the exchange rate, the spins cannot exchange with each other because now you are locking, you are holding that nitrogen from state A. Okay. Now you get another peak over here. As you keep going, your intensities go up, and effectively, as a function of CPMG spin lock field, your R2 effective can be plotted, and you can derive the information from this. Okay. This gives you, if you solve this equation, if you solve the fit over here, you can get actually not only the R2 effective here, but you can get the population chemical shift difference between the what you call as a low populated state versus high populated state. You can get the exchange rate, and you can derive very important biological parameters from that. Here is an example. We have got two very identical domains. One binds to RNA with two side exchange, and that's why there is a significant broadening by NMR. The other binds in a very loose way, or very weakly, I would rather say, not loose way. Now, the residues that are present in the first domain show significant CPMGs over here, whereas the same residues show a flat dispersion curve over here, suggesting that there is this motion, or these residues are flexible in this particular site that allows this particular helix to move over the length of the RNA. These are RNA binding proteins, and that's how you actually, we can predict that this particular domain slides over the length of the RNA, and whereas this other domain actually binds and goes off. So this is the power of this particular experiment. This particular experiment tells us that this is the motion, and this is not the kind of motion that is present in the other domain. So I will just, I have only 2-3 slides now. I will just close it in that. Okay. So that is one aspect of it. The second aspect is when you want to do discover the drugs, or you want to screen the drugs, you use something called as a high throughput HSQC screening. What happens there is each of these cross peaks that you see over here, as a function of ligand, and as the increasing concentration of ligand, shifts. Now, if you see here, the shift of this particular residue, or I think there is one more. Okay, no. Okay. The shift of this particular residue over here, there is no change in the line width here. Okay. The signal, whatever the width is here, or the signal intensity, what you see here, the signal over here remains more or less here. But whereas you look at this particular signal, this becomes broader in somewhere at this particular concentration, and then again it regained its position, right? Is it evident to you? And that happens because when it is binding to the ligand at this particular population, the bound conformation and unbound conformation of the ligand are able to exchange with each other, and that's why you have got a signal broadening, resonance broadening, and these are called as intermediate exchange. Okay, millisecond, microsecond timescale motions. Of course, as your ligand concentration enhances, you have got saturation, and then once you have saturation, you essentially come down to the saturated condition. So one can use this experiment very effectively to screen the drugs. Okay. Now there has been one more approach that has been very recently described, where in the very first experiment I have told you that there is a binding at site 1, site 2, but site 2 is dynamically not optimized. Now you optimize the site 2 dynamically by creating some kind of chemical stability ore in this particular case, and now you have done this by creating this additional hydrogen bond. In this case you can make any additional combinations in this case depending on the ligand by chemical modification, and now you are actually making it more stable, and that's how one can actually use dynamics in this particular case, make low dynamic so that the ligand becomes, the affinity of ligand becomes much better, and that has been shown in this particular case. The order parameter that I just described you, S square, essentially goes as a function of flexibility in this particular case, and it becomes rigid once the ligand is bound. Okay. This was another example where FKBP was, there was one ligand that fits into one pocket, other ligand fits into other pocket. Chemically they have more optimized it, and then eventually they created a new ligand which fits here with a very high affinity of 49 nanomolar or something. Okay. So there's a new, this is a vast landscape of drug discovery and role of NMR spectroscopy and particularly NMR dynamics into this. You have got multiple methods. There are a set of NMR experiments, relevant NMR experiments that you can see over here. As you can see there, I'm not going to read through it. There are some experiments like ZZ exchange or R1-Rho or even RDC, CES, DEST I didn't describe here today, primarily because they are more advanced experiments, have been discovered in the last 20-30 years, 20-25 years I would rather say. But they are very important experiments, and using all those experiments one can actually find out much more relevant information in terms of ligand protein interaction, and that makes people optimize the drugs a little better. So I think with this I again thank Neil for this kind invitation and bearing with my idiosyncrasies during the process. And I also thank some of the students who are partially responsible for generating this data. What I presented to you, this is our SOL 600 that we have at CCMB. This is my first student Chaitanya, who worked on 2 projects of RD4 and DR4. Rakhi solved the structure of CRC, which is 31 kilodalton. Upasana and Sneha is actually working on that artificially broadened domain that I told you. She found very exciting results. Right now she is communicating that. Jaideep, Ramdas, Devdatta and Preeti, they are working on different aspects. We actually look at something called as RNA binding proteins across various organisms and try to understand some very basic molecular biology, the properties of these pathways through structure and dynamics. Thank you so much.
Any questions? Thank you, sir. It was very informative talk.
Thank you.
Thank you for covering all the comprehensive aspects of structure and function relationship and how we can study that through NMR. I would request Dr. Neil to present a token of appreciation to Dr. Mandar.
Thank you so much. It's very nice of you.
Thank you. So I have a few.
Sure.
So I have few announcements because of the change of schedules today because of the meetings we had. So we will have only one lecture in the afternoon, 2:30. We will have Dr. Jithender Reddy will be speaking today, and then we will have Dinesh will take the practical part, and no, we will take the practical part. And tomorrow we have 2 important lectures. 9:30. Somebody from US, the Bruker has come out with a new instrument for metabonomics and biomarker discovery. So she will describe directly through the online mean, morning 9:30, and that is half an hour. And then immediately, so please be on time because she will be at the night time in US. So 9:30 we will start that talk, and then immediately after that, Dr. Neeraj Sinha will speak on some of the tissues NMR for biomarker, hair, bones, and other things. And then it will be mostly again experimental. So we really don't want to saturate you with most of the oral presentation. It's a more practical part. So that is the main reason. And thanks to Neeraj for agreeing to postpone his lecture for tomorrow. Okay, thank you. So you can go for lunch. We meet again at 2:30 for Jithender Reddy's talk.
awards. He has been awarded Young Investigator Award 2016 in International Conference on Magnetic Resonance in Biological System at Japan. Currently, he is working as scientist at CSR, Department of Chemical Technology, Telangana. Today, Dr. Reddy will talk about application of NMR in biopharmaceuticals. So, without any further ado, I would like to invite Dr. Reddy to deliver the talk.
Hello, can you hear me? Okay. So, thank you very much for the introduction, and I would like to thank Dr. Neil for giving me this opportunity to present something to new young minds. Today I am going to present, of course from the morning you were listening towards protein dynamics and stuff like that, drug interactions. So I would like to take a different little deroute from that topic. I myself, Jithender Reddy, I am working in CSR ICT, which is Indian Institute of Chemical Technology, which is a CSR laboratory, and our mandate is to have academic ambience with industrial outlook. That means we have an academic research going on, but support the industries, their problems and solutions. So this talk today, what I am going to present is something like that, which we are actually giving support to a lot of industries in assessing the biopharmaceuticals. So, I mean, in last decade, it attracts many of the pharma industries because there are a lot of funding is going into biopharmaceutical research because they are the ones now targeting highly chronic diseases. So today I will be talking about the NMR application in assessing biopharmaceuticals. Okay. So this is the outline. First I will just introduce briefly about biopharmaceuticals and biosimilars. This is the one now gearing up, and then I talk about what is biosimilarity, and then what is higher order structure, how NMR is involved in that, and what are the NMR challenges, challenges faced by NMR in studying the biosimilars, and then what are the methods to overcome the challenges, and then obviously there is a lot of statistical methods involved in this to assess the similarity between the biotherapeutics. Finally, I will give you a case study which actually used to get approved from FDA. So I take that as a case study and I explain how things are done in NMR to get approval from US FDA, which is a drug administrative organization that is used. Once you get the approval, then only you can place the pharmaceutical in US market, and they have several such guidelines are there. Like for Europe, they have their own guidelines, and for US, it is US FDA. So I will be explaining some protocols about this, and please feel free to ask in between if you have any questions. Okay. So first of all, what are the biopharmaceuticals? You heard about drugs, chemical drugs. What are biopharmaceuticals? These are a special definition given for this. These are the medicines which are the main component which is derived or produced from a biological source, maybe cells, tissues, or any of the living plant cells, biological source. So those the active ingredients in a medicine which are derived from biological source is called biopharmaceuticals, and that can be hormones, vaccine, blood products, allergens, many of them. Okay. So why these are so important? Because they exhibit powerful efficacy for the treatment of life-threatening diseases like diabetes, autoimmune disorders, and cancer, and so on and so forth. So these are the diseases which actually where chemical drugs are failed, I mean, to an extent, but the biologics or biopharmaceuticals are taking a lead role in addressing the diseases such as this. So what actually difference between a small chemical synthetic drug and between synthetic drug and biological drug is listed here, very crude way. The size of the molecule is small, and whereas biologic is very large, and structure is simple, single structure, whereas biologic will have a complex structure, and variability will be defined. Variability means batch to batch variability, whether they are every time they produce same identical copy drug or not. So that variability is highly reproducible. Like variability is very defined in small molecule, but it is heterogeneous in biologic because the experimental expression conditions, expression system, based on that the variability comes into biological products. So this is the difference.
And exactly how they manufacture this, small molecule is through chemical synthesis and biologics through living cells cultures. Okay, then characterization for small molecule is very easy. You can do complete structural characterization using NMR and other techniques. But for biologics, still you can do, but it is not that easy. Okay, and stability, chemical molecules are stable, but these are all sensitive to the conditions like buffer pH condition, temperature condition. They are sensitive and they can undergo degradation if you mishandle them. Immunogenicity, these biologics possess high potential of immunogenicity. So when it comes to specificity, this is the only game where biologics are taking over. Specificity is very high for biologics and which will help in treating this kind of diseases. Okay, so the special features why the biologics, pharma are looking at biopharmaceuticals they are very specific and there will be very rare side effects and they mimic body proteins, so it is easy to administer to patients unlike chemical drugs, and they have low off-target toxicity, and then they have long duration action, so less frequent dosage. So just like vaccines, you don't need, you cannot, just like normal drugs, you don't take vaccines, right? So you can take very, less frequently you can take these biologic dosages. But what are the limitations that the world is facing to produce biologics more than, still this is dominant. Nowadays this has picking up the momentum, but this still is dominant because of cost and critical storage and handling conditions. So these are the two issues which still bothers pharma industries when they want to jump into biopharmaceutical manufacturing or research and development. So there comes the biosimilars. These are the products which people are trying to synthesize with low cost and more, which will have more reachable to the patients. So they have framed a word called biosimilars. What are biosimilars? They are biopharmaceutical products highly similar to the reference product, which is biologic product. Reference product means the one which first innovator product, who invented the product, and after patent expiry that they take it as a reference product, and to that they always compare this biosimilar, the lab generated product. Okay, so the biosimilars are the highly similar to the innovator product with minor differences in clinically inactive components. For example, here if you see a big maybe monoclonal antibody or any protein which has a glycosylation sites, and you can play with different glycosylation sites which does not affect the overall structure and function of this monoclonal antibody or any protein, but still they are different from the innovator product. So they do little post-translational modifications so that they will have a different molecule than innovator product but have a similar structure and function. So biosimilar is something similar to reference product with minor differences in clinically inactive components with no clinically meaningful difference in terms of safety, purity and potency. That means there should not be any difference in terms of effectiveness, efficacy, safety and efficacy. So when these biosimilars can bring into market only after patent expiry of innovator molecule. So biologics or the innovator biological products are the patent is till 20 years. After that you can get into synthesizing biosimilars and you can leave it into, you can give it into market. So then this is a broad definition. What is the minor differences, allowed differences and allowed similarities. Here are some points to be noted. The similarities, these are also from US FDA website I have taken. These points should match with biosimilar reference product and these differences are allowed. For example, exact primary structure, therapeutic indication, that means where it exactly go and interacts and or the function basically, and the strength and dosage, route of administration, and biological activity, bioequivalence, purity, safety, potential, and mechanism of action. These properties they should possess. The biosimilars should possess same properties as innovator products, but they can have difference in minor structural variations, in formulation, stability, storage, expiry dates, and manufacturing processes, cell lines, purification methods, and their naming. Basically, they have a nomenclature. For biosimilars also they have certain nomenclature how to name it. Okay, so of course there will be a cost difference and packing and delivery kind of differences. So these are expected differences, these are expected similarities between biological product and biosimilar. I hope by now it is clear what is biologic and what is biosimilar. So then how to establish biosimilarity and what is the approval process for biosimilar products. So basically, so far we are talking about the properties, what to be similar and what not to be similar, but technically how you prove that they are similar or not. So basically we look at these things, comparability in terms of pharmacokinetics, pharmacodynamics, we say PK and PD, and safety and immunogenicity. Only these 4 parameters if you can understand, and these minor differences do not have a relevant impact on the final therapeutic result. So if you can prove these 2 topics, then you can get approval from the organizations where you can release it as a drug in the market. So to establish that similarity, biosimilarity between biologic and biosimilars and biologics, this is the process for biosimilars. You have a lot of work involved in physiochemical characterization where analytical methods involved, and then there are very few biological characterizations and even few preclinical characterizations and post clinical characterization. As you increase the data at the bottom, you need to work out on the top of the pyramid. Okay, so basically here just you measure PK and PD and safety. That is what all you do in this, and you always compare with the biologic. So you have a standard reference for that. So for doing the studies for regulatory approval, you need to know you should have thorough understanding of the reference product, because they have already put into market, so it is available in their US FDA website. You can go through the documents and you should be thorough of your reference product, then only you jump and work on your biosimilar to that product. So what are those characters you have to understand? This mechanism of action, structure-activity relationship, clinical knowledge, availability of clinical relevant PD measures, that is pharmacodynamics measures, and etc. So you should have thorough understanding of reference product, and then you start working on one by one. So if you think about a proper innovator molecule, so these are the document numbers in the US FDA website. So they refer this document number for biosimilar approval and this document number for originator biologic innovator approval. So in innovator molecule, it takes a lot extensive work in improving preclinical and clinical trials. So you have phase 1, phase 2, phase 3, so on and so forth. So you spend a lot of amount of time, money and everything in doing this work rather than this work. But when it comes to biosimilars, it is more of this work which is very easy and less of this work. That's why the biosimilars are less cost and it takes less time, and they are now all the industries are jumping into this and they are trying to put different biosimilars. I mean, in rough terms, I can say biosimilars are generics of chemical drugs. In chemical drugs, synthetic drugs, you say innovator drugs and generics. So here you say biologics and biosimilars. Okay, so now a lot of industries from last 5 years, they are working on different, because most of the biologics are patents are getting expired in these coming years. So they started working on building biosimilars on those molecules. So now I will go into this critical quality attributes. What are the qualities that you have to understand of biosimilar and compare that with biology. So these are the critical quality attributes. The most of the work goes in analytical studies and the very less in animal and clinical studies. So here if you see in analytical studies, you have most of it is structural analysis, where you understand primary, secondary, tertiary and quaternary structure of the molecule and post-translational modifications and any other variants or changes because of like deamidation, oxidation, aggregation and so on. So then you have to understand these 4-5 parameters in structural analysis and some functional assay like bioassays and binding assays and energy kinetics. So this all forms under the first category, the physiochemical properties, and this is studied mostly by the analytical techniques. Okay. So now I am focusing on this higher order structure, which involves all these 4 terms: primary, secondary, tertiary and quaternary structure of the molecule. So why this is so important? Because the higher order structure and dynamics of biomolecules are directly linked to their function. So from the morning 2 talks, you are mostly listening about that. The proteins and their dynamics are directly involved in their function, which is important. And if you want to target that function, you have to know the dynamics and structure of those molecules. So this higher order structure is directly involved in protein protein function. Okay. Then now what is the higher order structure, how we characterize, and any one method that gives high resolution information on this. So these are the two questions I will try to look at next coming slides. Okay. So to say the higher order structure is nothing but primary, secondary structures and tertiary structure and the quaternary interaction between the different proteins. So these all structure falls under the higher order structure, and we have to prove that is similar in both biologic and biosimilar. The standard definition is it's non-covalent interaction which stabilizes these structures, that is higher order structure in general terms, but it involves a lot of things. There is a quaternary structure, polymerization, aggregation, exchange, chemical kinetics, folding, unfolding. This all falls under indirect properties of your higher order structure. Whether the structure is intact or changing, these processes will tell us whether the structure is same or different. So indirectly we are looking at all these things in molecule and comparing that with biologic. So what is the importance of this higher order structure? That's the same thing which I was talking about. The proper three-dimensional structure is required for function, and any change in the structure will lead to low efficacy and side effect. And the structure and dynamics is related to protein stability, viscosity and aggregation. So these parameters are directly linked to its function and its availability and its efficacy also. So that is why it is very important. And again, same question, is any one method that gives high resolution as information on higher order structure? And yes, it is of course our NMR spectroscopy. That is a topic what we are meeting today here. So how this is involved in understanding the higher order structure similarity between biologic and biosimilars. So from last 3 days you might have working on NMR spectrometer and various things, and this entire topic involves only 1D and 2D NMR spectrum. You don't need to go think of beyond that. So here this is a simple 1D experiment where you have a relaxation delay and a 190-degree pulse and you acquire. So this is your real-time signal that you are collecting, which is called time domain signal, which is free induction decay, and upon Fourier transformation you get a spectrum which is a frequency domain signal. Frequency domain. This is a time domain signal, this is frequency domain signal, this is FID and this is spectrum. So why I brought this is, what is the parameters that you can capture from this NMR spectrum, how that is correlated to your higher order structure similarity. Okay. The first thing is peak position, that is chemical shift. Chemical shift is identity for primary structure, that is which amino acid it is, that means which atom it is, right? So the chemical shift says about the atom, that is primary, and chemical shifts also depends on the secondary structures. Based on the allowed sci-fi diagonal angles, the chemical shifts also vary. When that amino acid is in structured form or alpha helix or beta sheet or random coil, it takes different chemical shifts. So it also sensitive, the chemical shift is also sensitive to secondary and also tertiary, that three-dimensional structure. Okay. So is there any unfolding of this three-dimensional structure? You can simply see change in chemical shifts. Okay. The first property is chemical shift, and second property is the peak line width at half height. Even the talk which we heard just before, Dr. Mandar Deshmukh talk, he was explaining about the chemical exchange, where the line shape varies as the exchange rate increases and decreases. So the line shape also tells me about the interaction of molecules. That means whether it is undergoing any chemical exchange, quaternary interaction, whether it is interacting with other molecule or it is getting aggregated. All these properties, that is the quaternary interactions, and these things can be explained by looking at the peak width. Okay. And the last one is about peak height. If you take the intensity, area under the peak or peak height, that talks about the concentration and dynamics of the molecule. So all the important, I only left about one parameter, which is coupling constant, which is anyway we don't use here, but there are only 4 parameters you get in NMR, and you get enormous amount of information from those 4 parameters. Okay. So that's the beauty of NMR. So similarly, this is about 1D I talked about. If you have a 2D spectrum, you see peaks something like this, and it also gives the chemical shifts, it also gives the peak width, it also gives you the intensity. If you integrate entire peak, you get the intensity, and that will be your peak height. So you can apply same for even 2D experiment also. Okay. So Now, that's what you get from NMR spectrum. Now I use this 1D and 2D as fingerprints of the spectrum of molecule, fingerprint spectrum of the molecule, and then I compare 1D of both innovator and biosimilars and 2D of innovator and biosimilar. So here you can simply see this is the 1D spectrum and this is a fingerprint amide region of the protein, which anyway it's none of our interest because most of the proteins, folded proteins, you see something like this. So, and you see this is a 2D proton nitrogen correlation spectrum where each peak corresponds to one amino acid. So if you see that amide, of course this was explaining from the morning. So if you see amide, each amino acid will have one amide and you have all amides spread across the molecule. So these are my local reporters of the molecule. Any change in this, any locality of the molecule, then I can see a shift in these chemical shifts. So this HSQC, 2D HSQC is a fingerprint region for biomolecules. Okay. So just to explain for you, you see the same protein when it is folded, it is the chemical shifts are, the peaks are spread across almost 30 ppm in nitrogen dimension and 6 ppm in proton dimension. But when you unfold it, you see all are collapsed into a single region. So this shows that this fingerprint spectrum can also tell us whether protein is getting folded or unfolded. That means secondary and tertiary structures are formed or not. Okay. Similarly, we have other reporters that are methyls, side chain methyls. Just now in previous talk, Mandar explained that the methyls have a rotamer nature and they produce a very sharp signals. So when you go to high molecular weight proteins, generally you tend to get these signals tend to broad out. Then you cannot record nitrogen correlation. Then you go to methyl correlation spectrum where you can still take this as a fingerprint of big molecules like monoclonal antibodies, which are of 150 kilodalton size. So for these things, we depend on the methyl spectrum basically to look at the fingerprinting, matching fingerprints between biologics and biosimilars. Okay. Now I come to the actual work. So what is our aim in this is to establish the higher order structure similarity between a reference product and a new prepared biosimilar product. Okay. So what we do first, we record 1D, 2D NMR spectrum of both, and then we use some statistical methods. So basically for any NMR analysis, you undergo sequence specific resonance assignment. That means you have to identify which resonance in the spectrum coming from which atom in your molecule. That is called resonance assignment. For this, you no need to do such a robust exercise to record 3D experiments and do all resonance assignment. We just match the fingerprints. That means match the spectrum, one-to-one matching. There is no extra experiments record involved, no extra peak identification or something involved. Just you record the spectrum, whatever the information it gives, I have to just compare them. So for this, the blind thing what we can believe on is statistical methods. Okay. So the second part is to compare, just like fingerprinting comparison, we depend on the statistical methods to evaluate differences at atomic resolution using these spectrum. Okay. So first, what is the advantage of NMR experiments? They can detect change in all higher order structures and it gives a very high resolution information. That means any small or very relevant small changes it can detect. And in NMR you can measure the intact molecule in a physiological relevant condition, whereas in other techniques like X-ray you crystallize and do such things, right? So here in NMR you take direct formulation. That means you buy some drug from the pharmacy, if it is solution, just put it in your NMR tube, add some deuterium solvent and you record NMR. So there is no much of change in composition, change in sample preparation involved. So you can directly measure intact molecule in conditions that are physiologically relevant. And the other important thing is location of change in the structure can be directly identified. As I said just now, my amide protons or amide methyl groups are local reporters. Any change in the locality, they will show up in your spectrum. That's why you can easily locate the change on which side of the protein it is changed. Okay. So these are the advantages of using NMR in this, and NMR is the only one mostly now they are using for higher order structure analysis. And what are the challenges? There are few challenges definitely. The high concentration of formulation. If you want to compare the drugs when they are formulated, that means you will be adding some excipient to that to stabilize the molecule. These are called formulation. When you add these things, they will be very high in concentration. Here it is sorbitol, acetate. This is an acetate buffer, so you have acetate peak, and you have some water peak here. So in this, if you want try to look at the signatures of the molecule, it's highly impossible unless you zoom it 2000 times or so. So why? Because the dose is in low concentration. The actual drug substance, we call it as active pharmaceutical ingredient, is very less in concentration. So your actual molecule that you want to characterize is low in concentration, the other stupid things are in high in concentration. So it is very high, very challenging to record such spectra when you have some strong signals coming from some non-useful material. Okay, and again we have another problem. Generally, most of the structural biologists, they go for isotopic labeling. You all know that. Here, our pharmacy fellow does not provide us labeled drug products. It is in natural abundance. So I have to depend on the natural abundance 59 and 13C to record 2D spectrum. That is another challenge. And fourth thing is the molecular weight of the protein. So I mean, from last 3 days you might have understood that as the molecule size increases, the peak width or peak gets broadened because the molecule tumbles slow in the solution. In NMR, if molecule gets bigger and bigger, tumbling goes slower and slower and your NMR signals get broader and broader. So it is very difficult to study monoclonal antibodies. That's why in this we depend on the methyls most of the time. Okay, so to some extent to address this problem, USFDA always encourages to use 600 with cryoprobe, 600 and above with cryoprobe. So this is their kind of suggestion. I would not say that compulsory you have to use this, but 600 with cryoprobe and 600 and above with cryoprobe is good for look at these molecules and do higher order structure similarity characterization. So there are some techniques which I am not going in detail into that, but now we have methods, NMR methods, where you can use SOFAST. If you use SOFAST HSQC, you still get the HSQC correlation, the proton carbon or proton nitrogen correlation, but that will allow you to reduce the recycle delay D1 by almost 80% time. That means if you are spending like 10 minutes to record one spectrum, then your 80% time is reduced. You can record that in 2 minutes. Okay, so it allows you to decrease the recycle delay. So there is another method called non-uniform sampling, where they take only 50% or less than 50% in indirect dimension and they try to reconstruct back. That means you are recording 50% of the data and you are reconstructing the full data. So using these two techniques, we can make it very speed. You can record these spectra in very less time, and of course you can spend on the number of scans so that you have good signal to noise ratio. That is what we require because our drug product is very less in concentration. Okay, here is one example. This is a general HSQC and this is with SOFAST method. Okay, and with same scans. Sorry, this is same scans, and here the SOFAST method with 50% non-uniform sampling. So if you see here, instead of 8 scans, you are now allowed to run 64 scans within same time by using SOFAST method and NES method. So that's what I am saying. You can reduce in time, but you see signal intensity gain because you are recording much more number of scans here. So using these two techniques, you can get high intense spectrum, which is very useful for your statistical similarity correlation of biologic and biosimilar. Okay. Then the second problem is with the excipients. They have very strong signals coming from excipients. There are some techniques called Sierra. So all NMR community is very fond of giving fancy names to all their developments. So these two are such their names. Sierra is nothing but it will selectively suppress the excipient signals. You see very strong excipient signals here. This is the signals coming from my compound or drug molecule, and these 3 or 4 are coming from the excipient. When I use Sierra, you see the decrease in intensity of the excipient, and there is some algorithm where it can even subtract further those strong signals. Using that algorithm, you can completely remove excipient signals without any altering any signals coming from the compound. So these are the two techniques recently developed for suppressing the excipient signals. And one important thing here is we are not losing on any signal to noise ratio. Generally when you do this kind of editings, you always end up losing some signal to noise ratio. That means your signal of your compound also goes down or decreases. But here I have compared all four, in this paper it is compared all four different setups, and you see signal to noise ratio is almost intact. Okay, then the last one is about the molecule size. As I said, as the molecule size increases, you will be seeing a broad signals in your spectrum. So how to come out of this limitation? There are two ways people optimize this. One is either you record at high temperatures, but in our morning discussion people say that biologists, they don't want to deviate from physiological conditions. So you have to record at 37 or 25, somewhere like that. Maybe for them this choice is not applicable, but still people try to record at high temperature on both the reference product and biosimilar product, and they compare. And second point is, especially for monoclonal antibodies, there is something called papendization. It directly diates between this FC fragment and a Fab fragments, and now each fragment acts individually as single molecule, and they fall under very low molecular weight, not very low but 50 kilodalton, which we can record through NMR. Okay, here, okay, for as far as temperature is concerned, monoclonal antibodies are very stable. Their actually unfolding or thermal unfolding temperatures are around 60-65. So you can safely go up to 50 and record that. So that is acceptable for even US FDA and other regulatory bodies. So you can in principle go at 50 degrees and record this full monoclonal antibody spectrum, or you can use papendization, where you can digest all the fragments and you can still see that the blue one is intact, spectrum of intact monoclonal antibody, and the red one is the digested fragments Fab and FC maintained in 2:1 ratio. I recorded on that one. You still see there is no chemical changes or there is no environmental changes in these peaks. Maybe 1 or 2 peaks, but mostly it is intact. Okay, so these are the two techniques now people are using to look at monoclonal antibodies and characterize their higher order structures. So this is about the techniques, advanced NMR techniques that you can use to record the spectrum. Now we go into statistical methods. What are the statistical methods useful to compare these two spectra? So in around 2016 to 18, these are the proposed methods, but very extensively used. And one is the visual inspection, where you directly overlay and just send the spectrum to FDA and they look into it and they get the information from this. That is visual inspection. Second is combined chemical shift deviation. Third is intensity correlation plots, and then the fourth one is multivariate analysis, which is PCA, principal component analysis. This is very well used in any of the statistical analysis, even in metabolomics, and metabolomics are all people are using this. So we use same technique to identify the similarity between biologic and biosimilars. In combined chemical shift differences, what we do is we take randomly starting from the left top corner or right top corner to left bottom corner, we try to take the chemical shift of all the peaks in systematic manner in both spectrum, and we try to see the differences. Just like RMSD, we try to see the differences between the peaks which are almost nearby, and we take those two peaks and we try to see the differences between those two peaks in two different spectrum, and that we calculate as CCSD, which is combined chemical shift difference. And if it is less than this, and it is allowed that these two molecules are similar. So if it falls 0.008 ppm, that means 8 ppb, parts per billion. So if it is less than 8 ppb, then it is allowed. So this is the standard they have established, and you simply take all the peaks from one corner to other corner in both the spectrum and compare one to other and see the chemical shift differences between those two spectra, and you end up plotting this correlation. The third thing is, the name again gave a fancy name called ECOS. It's nothing but comparing the intensities. So easy comparability of higher order structure, that is ECOS. So it is just you bin entire two-dimensional spectrum of biologic and biosimilar into several small bins. You take those intensities in those bins and you try to correlate one with other. If the correlation quotient is above 98, then this is allowed as similar compounds. So just without doing rigorous NMR assignments and all, you just take the spectrum, you compare. These statistics are very well established in most of the data analysis. So here they use this to do higher order structure similarity analysis. So to get into more robustness and to understand this characterization in more robustic way, there are several labs involved across the globe, almost 26 labs involved to characterize monoclonal antibodies using almost 375 spectra, 7 different magnetic fields, 39 magnets. So they try to get as much as statistical data and correlate whether these limitations or whatever I was talking about, the cutoffs, are they really correct or true or useful or not. So there are 26 labs come together to measure these almost 375 spectra, and they try to, there is a discrepancy here and here. Anyway, so they try to do analysis in both CCSG and PC analysis. You can clearly see at different temperature they are segregating into different regions. That means you can still use PC analysis to understand the similarity between the products developed by different labs and recorded at different magnets. So you can still, using PCA, you can correlate whether the two molecules are similar or different. So this will tells you about the robustness of the chemometric analysis here. And this one you see the same spectra are used to calculate the CCSG that I was talking about, and all of them are falling within the limit that was set. Okay, only these two are falling little, having more error because they are actually corresponding to non-uniform sampling schedule. This one, the spectra which are non-uniformly sampled, maybe the sampling and reconstruction of the spectrum is not that great. So that's why you have long errors here, and also SOFA 6 MQC where these two are showing little more errors, but otherwise it is very well within the limit. Even this PCA chemometric analysis says that you can still segregate the molecules using PC analysis done on NMR spectrum. Okay. So now I would like to go to case study. So I have pasted here some of the queries came to the labs which we are collaborating. I am not going to name the labs and the compounds, but I just want to show you that what is their requirement, FDA requirement. This is posted to one lab. Here they are asking to establish hierarchical similarity, following points must be addressed during the analysis using 1D proton 13C correlation, proton 15 correlation. With 1D NMR spectrum, they are asking for principal component analysis using a Mahalanobis distance calculation. This is the famous mathematician from Kolkata, India, developed this. He come up with some equations which can actually calculate the similarity distance between two clusters or cluster to a particular point. Okay, so they are asking to go with principal component analysis and Mahalanobis, DM calculation. So, and they are giving one reference also in this. Directly they are giving, you do as it is done in this paper. Okay. And second thing they are asking is to do spectral comparison. They are not asking PC and DM calculation on 2Ds, but they are just asking for a spectral comparison of drug and test products. Okay, to compare the to calculate the HODs. So here in these two spectra, they are asking for peak height comparison and chemical shift comparison. Okay. So this is the lab 2 from FDA. Same question. Proton NMR spectrum, 2D HSQC and 15 SOFA spectrum, and you have to do principal component analysis and to get the similarity. Again for lab 3, this is exactly quote unquote, they just copied and pasted into our mail. So with quotes, your response was reviewed and your findings of NMR spectral difference caused by low acetic acid. So here actually this lab produced their biosimilar different pH, not the same as a reference product. So here they are asking little extra data saying that caused by your findings of NMR spectral difference caused by lower acetic acid, higher pH and more aggregation in exhibit batch were acknowledged, but the higher order structure similarity between your test product and RLD remains to be demonstrated. You may report the protocol of pH titration, collect 1D NMR spectra on pH titrated test product and RLD for chemometric comparison using this paper. So he is asking them to prove that wherever your reference product pH and your pH, I want to know that in both states the both proteins are similar. So these people come up with 4 different pH in that region, and they gave us the samples, and we did the chemometric analysis for those four different set of samples. So always, this is very recent, almost whatever I pasted here, 3 labs in last 1 year. Okay, and they always quote these 2 papers. In this, the entire protocol is discussed, and they want to exactly reproduce the higher order structure similarity if they are similar. So in this paper, I just go through the paper because I can't produce my data, which I have done to the labs, it is confidential. So I am going through the data published in that paper, and I try to explain what they have done. So here in that paper, they have taken 6 different biosimilars. Most of them are kind of peptide to protein molecules, and one is monoclonal antibody. In this also you can see this is very well structured protein with very low concentration, and here is a good concentration, but the protein is undergoing exchange or aggregation. You can see a broad peaks here. Here you have all sharp peaks because it's just monomer and freely floating in the solution, and this is undergoing exchange. That's why you see a broad peak. They took different molecules having different properties. I said HOS can characterize the primary, secondary, tertiary, quaternary, aggregation, folding, blah blah blah. So it can characterize all these things. So that's why they took all different molecules and tried to establish the statistical correlation with this molecule. So here this is a undergoing aggregation. These 2 are the proteins undergoing chemical exchange and aggregation, and the one you have monoclonal antibody. So they directly recorded 1D NMR spectrum of monoclonal antibody. You can still see intact monoclonal antibodies, 1D proton 1D NMR spectrum. Okay, so these are the ones. It's a full spectrum with all excipient signals. You don't see any drug product signals, but when you zoom in, you can see all the drug product signals here. So they have studied all of them, but I'll go only using insulin product. This is the one they used to establish the cutoffs, the limitations to judge whether it is similar or different. So here insulin molecule, this is the primary sequence of insulin molecule. It has 2 chains, chain A and B, and upon certain change in pH, it undergoes dimerization, and with different excipient like phenol and zinc metal ion, it forms a trimer. So they have taken all different insulin products available in the market, which are actually approved across almost 3 decades, 82, 91, 96, 2000, and these ones are reference products, biologics, and these are their actually biosimilars. All are insulins but produced from different organisms. You see human insulin, insulin glargine, insulin lispro, and they have different mutations in their biosimilar product. So they took these 6 set of things, and they have different action also. One is short acting, one is long acting, one is rapid acting. So they took these 3 different batches.
First rapid acting. This is biosimilar because I think.
No, actually in earlier days there is no concept of biosimilars. All are biological products. That's why this is a different molecule. This is biologic.
Okay.
And this is also biologic, a different molecule. Expected from
what
different
represent then N2K?
These 2 are in this one. This is the biosimilar for this one.
Okay. For in Basa color
Yeah.
They have made the mutation
Yeah.
N2K in the
Basagar is
lentils.
a product name of biosimilar
Okay.
of insulin glargine.
Okay. So the second one is the biosimilar for lentils.
This one is a biosimilar for Lantus.
Lentils.
And this one is biosimilar for
for
Humulin.
this one.
And this one is for Humulin.
Okay.
Yeah.
Okay.
Okay. So there are 3 set of reference product and 3 set of biologics of insulin extracted from various sources. Okay. And they try to establish the correlation between this. So recently,
All three are biologics.
yeah, the first ones are biologics, and the last ones are their biosimilars. They are generics.
In general, this in the first row,
Huh.
both collective they are known as biologics.
Okay. No, here I am not talking about what are the, see, this is the molecule, API, okay. In this API they made these mutations to bring out
Biosimilar.
Yeah. Okay.
But in general, what is this term used for this? You are saying biosimilar then biologics. These in general these are biologics,
Okay.
including
So
biosimilars.
this one
Huh.
is API, drug product.
Yeah, but
Okay. So you put it in different formulation, it will become different brand name, and it goes as different. So okay, innovator will define entire thing, formulation, production, everything.
Okay.
Any deviation from that, you take it as a biosimilar.
Biosimilar. That
Yes.
is the understanding.
Yeah.
But what are then biologics?
Biologics are the first ones, innovators. There are biologics.
So then humulog is not biologic. It is a biologic.
It is biologic. This is
that's
biologic.
right. That is a biologic. Okay.
But this is the active ingredient. It will be presenting in both.
Yeah, that's okay.
इसमें बस this got mutated
Mutation.
with this.
Then that is why they are saying that biosimilar.
Yeah.
Okay.
Okay. So they took all these 3 sets of reference and biosimilars. Then they went ahead to look for the similarity matrix. So there is a lot of course, this is the regularly used for any metabolics or statistical analysis who is using NMR data. But still I go through what we do in this and what is that defined in the paper to use. Okay. So first you have to do the data processing obviously, and the data processing should be uniform with uniform parameters, I mean identical parameters. You have to process entire data of biologic and biosimilar. Recording and processing should be in with identical parameters. Second important thing is trimming. Trimming means this is a process where you exclude unwanted signals that are coming from the excipient. So then we remove the excipient signal from the data. Second process. Third is binning. Here we reduce number of variables and minor artifacts. What I mean to exactly is, for example, when you take a 1D spectrum, when you process it, you give some 30K or 60K points, right? So that is a digital resolution. So your each spectra is built by points with 60,000 points, okay? And it has maximum, it has noise in it, and very few points will have signal in it, okay? So to remove the minor noise artifact, what we consider as 0.02 ppm above, we take each 0.02 ppm, we take it as one bin, and we sum all the intensities, all the points in that bin. That is called binning here. We use some binning method, and here the allowed is 0.02 ppm and above, because your proton or your spectral resolution maybe falls below this. I mean, below this is not significant because your spectral resolution is less than that or more than that. So generally we have to bin. So this will reduce the variables also. It is easy for statistical methods to analyze. So for example, if you take 32K, when you try to bin with this and you are recording from almost -2 to 15 ppm, if you bin with this value, you end up around 200 to 300 points. So you have very less, 30K to 200 points. Use that 200 points to do further statistical analysis. So the third method is binning. We bin the values into small bins. We take the intensity values into small bins and then we do sum of all those bins. So this is a mathematical equation what I just kept here. So after that we do normalization. This is very important when you try to compare different spectrum, because they are handled at different concentration, they are handled at different temperature, different spectrometers and all. So to normalize the experimental variations from the biological variations, it is important to do the normalization. So you normalize your intensities with respect to the some parameter in that spectrum. Okay. Here we actually normalize over sum. So we take the summation of entire intensities and we normalize over that sum to bring into small values. So that is very useful for do quick statistical analysis. So here normalization, we do sum normalization, which is, you can see here, we take the summation, squared, sum, and then again under root. So we take the summation of all values, and with that we normalize our each individual value. After normalization, it is scaling, which is actually very useful to increase the required information. What I mean to say is, the signal you want to see large values, important values, then the scaling is the method to decrease the relative importance of large values and in data, keeping the data structures, and it is sensitive to large fold changes. That means you will still keep intact the large changes and keeping removing the minor artifacts. So here we use mostly Pareto scaling, where the data is mean centered. That means you always subtract your value with mean value, and it is scaled with square root of its standard deviation. This is standard deviation of these values, and we take square, and it is actually scaled with the square root of standard deviation. So this is the Pareto scaling. We use Pareto scaling to get the final meaningful data to do PC and DM calculations. So these are the process involved in treating data pre-treatment before doing PC analysis and DM calculation. Okay. So after that, this is PCA. I no need to explain. So PCA is a statistical procedure to transform a set of correlated variables into a set of linearly uncorrelated variables. Here, for example, it is completely non-correlated, correlated variables, and it tries to look for make it non-correlation by fitting it to the correlated axis. So now the new principal components are your correlated axis, so that the data points with respect to that correlated axis, they will become uncorrelated. So by this method, you end up calculating the principal components. Okay. So and we use those principal component for calculating the Mahalanobis distance. So as I explained, Mahalanobis distance is a measure of separation of data sets. Here you can clearly see it will measure the distance between the centers of data set. That is the mean center of these two data sets. It is nothing but the distance between these two sets. Okay. So if you have enough number of data points, you can have a 90% confidence helix to that data point. If the drug product falls within this confidence limit of reference product, then we say these two are similar. Okay. That directly you can visually also inspect, and also you can calculate through Mahalanobis distance, and you can put it as a unit, as a number. Okay. So here this is the one recorded and calculated the PCA, did PC analysis and calculated DM value for Humalog and Admelog, which are actually approved in 1996 and 2017. So those two are actually insulin Lispro products, and they all fall within 1.58 similarity distance.
Reddy,
So,
what is the cutoff?
I'll come to that.
Go to first previous slide just for this. So green ones, this is your blue ones are your
Yeah, these are reference data.
reference data.
These are a schematic example.
Schema.
Yeah.
But according to this figure, green ones are
Allowed. Similar
by similar,
similar.
but they are falling out.
Yes.
That means that does not fulfill the criteria.
Yes.
Okay.
So here, to do this analysis, one requirement is you should have more number of samples from same batch. Okay, with single reference product and single biosimilar product, you cannot do this statistical comparison. So the more number of points, you will get better confirmation. So here, between these two products, it is highly similar. Okay, And when you go to the other product also, insulin glargine, which are accepted in 2000 and 2015, also falling in the criteria with DM 3.29. So when they started recording on the old samples, which was 1982 approved and 1991, which is the human insulin samples, so they clearly fall apart. Okay, and these two are not similar, but they got accepted much earlier before putting this criteria on. So this separation is maybe because insulin human products were independently approved in these 2 years, and they are of different expression organism or production purification formulation differences. So there are entirely differently expressed, and the process is different. That's why they say that they are not similar. Okay. So the same calculation, they took a monoclonal antibody and they recorded 1D on monoclonal antibody, and they tried to calculate the PCR, do the PCR analysis and DM calculation, and they fit very well within the low values, and they still correlate with each other. So they try to see the field dependence also. You see 600 and 850, and there is little high DM in lower fields because the spectral resolution, obviously. So, but still it falls within the limit. So using these numbers, they try to. Oh, sorry, where is? Okay, I will. In one slide, I'll explain all the limitations, what are allowed values for all these things. Okay. Now I have explained about the 1D analysis. Now we move on to 2D analysis. There is not much to do in 2D analysis. Simply, this is the methyl region of two products overlaid. One is humulin and an homologs, red and blue. And here also the methyl region of this. Here, whatever peaks you are seeing is coming from the methyls of alanine, valine, isoleucine, and leucine, and threonine. So from these you get the peaks. You randomly identify them and try to calculate the chemical shape difference. Just like CCSD, we do individual chemical shape difference for both, and we compare them. So here the chemical shape difference between two reference product and biosimilar. And remember, this is in, please see, this is in ppb, not ppm. So 8 up to 8 ppb it is allowed. So they are falling within that region in both proton dimension and carbon dimension. So this is the 2D analysis. Here each individual chemical shapes are taken from the average of 5 lots, because they have provided 5 lots of reference product and 5 lots of biosimilars. These chemical shapes are taken as an average of all the values from each 5 lots. Okay. So the next thing they have compared is the intensity, relative intensity. Here they took the peak which shows highest intensity. Here the threonine, one of the threonine is showing the highest intensity. So they took that as 100%, and they relatively calculated all the other intensities in both in reference product and in biosimilar, and they try to correlate the intensities, individual peak intensities here in this one, and they calculate the p-value, which is the statistical, which will say the statistical significance, whether they are similar or not. So if it falls above 0.05, that is similar. If it is falls below 0.05, it is not similar. So if you see, out of so many 48 peaks, only one peak shows it is not falling in the allowed region. So here they come up with more than 92%. If it falls in this allowed value, then they consider it as similar. Okay. Here they're summarizing all this thing. If this similarity distance DM falls below 3.3, then they accept it as a similar compounds. When it comes to 2D spectral comparison, if proton falls less than 4 ppb and carbon falls less than 15 ppb, they allow it. They call it as similar, and with 98% comparable peak heights, these are the limits they gave to consider whether it is biosimilar or not. And this entire thing is discussed in this paper. So this is the way we report. One of the molecule, leuprolide, we calculated. This is the RLD spectrum, and this is drug product. and the shaded regions are excluded regions because they contain either noise or strong excipient signal. So we removed this and tried to calculate the PCA. We took 3 principal components, but the first component itself explains the 90% variance, explained variance. So the first one itself is, so even in calculating the DM, they look at the PCs which fall more than 90%, 90 explained variance. So here first principal component itself is explaining my more than 90% variance. So they take this value and they consider this as DM value, and they, if it is falls below 3.3, they accept these two as similar. So here I produce them, what is the bin resolution I have taken, and what are the number of bins finally produced, and the number of PCs calculated, and their explained variance in percentage, and what is the DM value. You can calculate DM value using only one principal component, 1 and 2, 1, 2, 3, all 3 together. So by looking at all 3 also you have less than 3.3 DM. So these two products are similar. Okay. So we give conclusion something like this. From the PC analysis of NMR data concerned 2 drug classes RLD and DP, it is observed that first one PC could explain 93% of total variance in the data and resulted in a DM value of 1.2, which indicates that the 2 drugs are similar according to this reference. However, analysis with a greater number of samples results in more statistically significant conclusions. So this is what we give as report to them to submit to USFD. Okay, finally, that is the conclusion. In workshop I always quote this because I am a fan of NMR spectroscopy, because there is hardly any technique that is so informative for so many different types of applications, and because no other technique provides so much fun. So only certain set of data, but as you go on digging for the information, you get a lot of information. And this is a quote from the great Nobel laureate Richard Ernst, who passed away last year. He got Nobel Prize in Chemistry for developing multidimensional techniques. Thank you.
Thank you, sir. Any questions?
Hello, sir. Yeah, nice lecture. So, like in some case, you have higher order structure, but the active is some monomer. Yeah, like say I'm taking an example. So like we have higher order structure, maybe some hexamer or any oligomer, but the active structure, active form is monomer. That after taking, so only monomer will work. And somehow in one case, someone has shown that monomer is active, that monomer is working fine, but higher order structure is not similar.
Okay.
So then, what's the use there?
So far we discussed about the top analytical portion. There is PK, PD, efficacy and all. So that also plays role in approving a drug. So if they are satisfied, they try to re-look into analytical methods again. Okay. And they will address little more. And second thing, I have to say whether one-to-one comparison, if it's same or not. If the property is same with the reference product, then obviously that is USFDA went through that thoroughly and they approved it. So that they don't take another chance of again doing entire thing for biosimilar. So if it is not falling similar with reference product, then definitely they want to take it as a complete new study, and they take it as complete new, like originator studies or innovator studies. Yeah.
Thanks.
HPLC profile also has to be really matching so that you don't have any minor contaminant of anything. Second thing, to also want batch to batch mass spectrometry profile also. That gives the mass distribution. And then for higher order structure, the NMR. And in some cases, they want crystal structure also. Okay, so all 4 things, analytical tools are required as far the FDA is concerned. It's not that only one they will say, oh yeah, it's fine.
HPLC chromatogram also should match.
Yeah.
So if there is any oligomers or aggregation, it It should be there in different products.
So if you go to in India, I mean, most of the companies like probably we can name few of them like Wokhard is doing, Biocon is there, Stelis is there. All of them actually batch to batch, they get this all this thing confirmed, all of this.
If you allow 5 minutes, I want to show direct analysis here. This one. I can't see. I think this one. Anyways, oh sorry. So this is, I mean, there are commercially available softwares from MNOVA and Bruker, but this what I want to show is that what I have coded with Python directly, and we do analysis in this and we produce. So we are not spending even a rupee to buy a commercially available, because nowadays Bruker also cashing on these biosimilar studies. They're costing for, like, if you want to, first thing, I think the CSIR ICT NMR lab is the only one research lab, government research lab, US FDA audited, because we do a lot of these studies. As a part of their auditing, they come to our facilities also, and they inspect our facility, and our facility we should maintain in regulatory environment, that means GLP and NABL regulated environment. So what all went into that? I mean, maintaining the machines and maintaining the things. If you want to ask, if you go and ask Bruker, they cost like anything. They will cost you for everything, software and everything. So we, with a small software team, we try to develop our own software so that we can do our own analysis, not depending on any commercial people, so that we pay them, and a lot of time goes into paying to that software. So here simple, I mean, it's still on the preliminary thing. So this is the data that I have showed. We have almost 6 RLDs and 3 drug products. I kept it in the data folder, just 1D spectrum, nothing else. And first is directly one script where I give phasing, and this is parameters are well set in this except phasing, because every time I take different phase. So except phasing, they all parsing parameters are set in this program, and just simply run this script. Oh, there is an error. Anyway. No, I have not built up to GUI yet. Actually, there is no software team, my and my student, that's it. There is no big software team to say. Okay, sir. Because of resolution issues. data, and it is processing with 32K points, you see here. Okay, and okay, I'll delete all of them. After that, directly it will display FID and spectrum. These are the processing spectrum of all 6 data sets. And after that, I just take one file for data trimming. In this, I have defined all the areas that has to be trimmed in this file. I can show you that. Something like this. What are the ranges that you have to exclude? Okay. Then it will do data trimming. After trimming, you see it has come down to 14,000. From 32,000, it has come down to 14,000. And after that, you have another script for binning and normalization and scaling, both, all 3. Okay. So after scaling, you see that has come down to 500. So now we are using 500 data points which are binned, which are normalized and scaled using Pareto scaling. And that file you can use directly as PC input file. Okay. Then the last one is PCA fitting and DM calculation. So here I can define how many PCs I want to calculate. So just run this. These are the explained variance. If you see 57 in PC1 and 33 PC2 and 8 in PC. So if you add all these 3, roughly it comes around 58. It comes, first 2 itself is giving more than 90%. Okay. So, but including all 3 also, you are getting DM less than 3.14. Okay, and here are the fittings which it are directly generated. This is one PC1 versus PC2 correlation, and this is another one. So here there is only 3 and 3. There is very low statistical data. So okay, so you don't expect very beautiful full round overlapping things. So it's only recorded with 3 RLDs and 3 reference products. So like this you can directly, so far I was giving almost half an hour lecture, but when you write it into a script, it's just a half an hour there. So I'll give my student all the samples to record the data, and both sit together. I do this in 5 minutes and I get good payment from industry for writing this data. Okay. So just to motivate you towards this. Thank you.
Any question?
One suggestion is nowadays research, it should go with multidisciplinary. So just learning NMR, just writing or PowerPoint presentation doesn't work. One should know how to do coding, because the next generation is completely on computer coding and data analysis and all. So it is good to have multidisciplinary approaches to look at any problem.
Thank you, sir. It was really a nice and descriptive description how we use NMR in pharmaceuticals.
Thank you.
Now I would request Dr. Neil to give a token of appreciation to Dr. Reddy.
Thank you. That's heavy.